No. Please go ahead.
गोल्ड मीट लगा ओपन में चलूँगा गोल्ड मीट 11 मणि ओके फस्ट मीट ओपन मणि स्टार्ट यार वो फस्ट तिरुमले का गुड name sir cashew
Good morning to one and all present here, respected delegates and participants. We shall now start the third day of the seminar, Indo-French seminar, chaired by Dr. Sebastian Kunak, IRD, Montpellier, France, and Dr. N. Geeta, Bharatiyar University, Coimbatore, Tamil Nadu, India.
Good morning. Now I invite Dr. P.P. Thirumayaswamy, ICIL, MB, PGR, registration, Thrissur, Kerala, to deliver his topic.
Respected chairperson and distinguished scientists, very good morning to all. At the outset, I sincerely thank the Indo-French organizing team for for inviting me to deliver the topic on sesame phyllodes, challenges to the sustainable oilseed production. You know, sesame is one of the important oilseed and ancillary food crop belonging to the family Pedaliaceae. And being originated in India, we have very good genetic diversity in the sesame indicum. And the top sesame producing countries are India, China, Myanmar, and Tanzania, which contributes nearly 80% of the sesame productions. Besides, we have wild relatives in India, and they are Mulayanam, Prostratum, Malabaricum, Laciniatum, and there are some species which are introduced as wheat species and got naturalized from Africa. They are the Sesame alidum and Sesame radiatum. The major constraints in the sesame productions are starting of the seeds. Besides, there are abiotic stresses such as waterlogging and drought contributes major constraints for yield losses, and biotic stresses such as insect pests and diseases also causes considerable yield losses. Prime among them the phyllodes, which is caused by the phytoplasma. It is reported from nearly 20 countries, but not yet reported from USA, China, and Australia. India, the yield losses ranges from 30 to 75%. In Myanmar, it goes up to the 90%. The earlier the disease reported means the losses increases. In average, in one of the experiments says that every 1% increase in the disease incidences, losses goes to 8.5 kg per hectare. This is caused by the phytoplasma. This is a prokaryotic molecules without cell wall and having the single membrane, so it is a pleomorphic nature. It is a phloem bound and it moves to the phloem cells, multiplies and causes diseases. They transmitted by the insect vectors, in this case the jacids are the major players. The array of symptoms are produced in the infected plants in the sesame indicums ranging from stunting and yellowing. It is the primary symptoms in the early stages. In that case, there won't be any fruit forms. In the later stages, if it infects, it causes witch's brew, phyllodes, virescence, flower bud proliferations, proliferation of axillary shoots, little leaves, leaf curl, and purple leaves. Again, seed germination, splitting of the capsules, and shoot tip fasciations like that. It is not only produced in the sesame indicum, but also it is a host for many wild relatives of sesame indicums, namely the Malabaricum, Mulayanum, Prostratum, Laciniatum, Radiatum, Alatum. I think most of the wild species, whether it is a native species or the species which are naturally found in the African countries, if we introduce in India, they also become susceptible to the phytoplasma. This sesame prostratum, which is a coastal bound, it is just 50 meters away from the sea waves, naturally found in the coast from the Visakhapatnam to the Kanyakumari area. In the patches, it founds always in the patches of thousands, and equally the plants are infected in the ranges of 15 to 25% in the case of prostratum. The characters which of the prostratum is deep-rooted and succulent type, and it is perennial type. And the capsules are indehiscence, and it is growing in. It is a salt tolerance because in the seashore it grows. So some of the traits we can use for salt tolerance or indehiscence type, we can take the traits from this prostratum. But the species is susceptible to the phytoplasma causes phyllodes. The other species, which is very similar to the prostratum, is another Laciniatum. It is found in the hilly area of small hills, ranges from 10 meter to 100 meter above mean sea level. In the rocky provinces, it also the deep-rooted system, drought tolerance. The capsules are indehiscence type, but this species is also susceptible to phytoplasma. The other two species are Mulayanum malabaricum. It is very close to the cultivated type Sesame indicum, and it is equally susceptible to phytoplasma disease. Then another two species which are introduced and naturalized as weeds, we found mostly in the roadsides, this Sesame alatum, the pink color flower plants, whereas the other species Radiatum, which we found in the northern India, starting from Maharashtra, Madhya Pradesh, and apart from Delhi, Haryana. And these This species also equally susceptible to radiatum, but alatum is the least infected. Among the 6 species, the alatum is the least one. We found only maximum of 6%. One advantages of the alatum is drought tolerant, and it is a transplantable crops. That means we can take from the native place and we can transplant in some other places. There is more than 90% success of transplanting will take place. For breeding purpose, we used to take seedlings from here and we used to plant at the Epidemics Trishur, and we will use for crossing program. But there is some seed biology plays major role. It won't germinate in new niches, means it needs such a dry places, less rain, such a conditions only the plant germinates. And this plant is having short duration. That means it completes the entire life cycle within 60 days. So one advantages we can take, one is phyllodes tolerant or phyllodes resistant, and a short duration it matures. So these are drought tolerance. These are the traits associated in the species, but less infected by the phytoplasma among the several species found in the India. Not only the native species, other exotic species when we import for a breeding program, that also become susceptible. The Sesame angolens, angustifolium and latifolium, which are found in Africa, and we imported it, also become susceptible to the phytoplasma diseases. So inside the phytoplasma, inside the plant, the phytoplasma moves through the phloem tissues and multiplies and causes diseases by manipulating the DNA of the host plants and produces symptoms like phyllodes and most unobservable symptoms like zygomorphic plant completely transform into the actinomorphic like plants. We cannot alter such a plants, very difficult naturally. So these phytoplasma does the job of engineering the plants to change into the zygomorphic to actinomorphic plants. The infected plants produces the phloem necrosis, and ultimately the plant dies. So the transmission takes place naturally by the insect vectors, leafhoppers, and mostly the jacids plays a major role. The insects are phloem bound, and second one is the dodder, which we will use normally for transmission studies in the laboratory conditions and also for pure collection, pure isolation of phytoplasma. And grafting, which we will use for the transmission studies, whether the plant is resistant or susceptible, we will use the grafting studies besides the insect natural infestation by the insects. Then the phytoplasma disease is, the phyllodes is not transmitted through the seeds. We have also studied, and early report also says that there is no seed transmission of this disease. Coming to the vector, the leafhopper, the Orosis albicindus and Hisimonus spicetis are the major one. In India, both the two species are reported as a major, and some other countries reported third species also. So we already discussed it is a phloem feeders. So it transmits the insects, saps go along with the phytoplasma goes inside the malpighians and guts, and the salivary glands, it multiplies, and it transmits throughout the entire life cycle of the insects. So this is the problem of the phytoplasma acquired vectors. Then one of the important one is that the nymphs and adult acquires, and adults transmit the entire duration of the life cycle. The acquisition feeding period, inoculation feeding period, everything is given. Minimum 30 minutes is sufficient to transmit the phytoplasma successfully to the healthy plants. Coming to the classification, what kind of phytoplasma associated, there are 5 groups of phytoplasma and 9 subgroups were reported as far as throughout the world. Among that, the 2 groups are existing in India. One is 16SR1B, another one is 16SR2D. These two strains are mostly found in the phytoplasma infected sesame plants, and we have also found. In the notable thing is that even the same field, we have grown different species, let's say Indicum, Malabaricum, Mulayanum, Radiatum, and in one group we found only the 16SR2D and 16SR1B in the Sesame Indicum. We did not find any mixed infection, and we did not find why in the same class the two exist, and and one group of phytoplasma infecting one kind of plants we do not know. We need to study more on that. It is still in the incipient stage. We have to do more research on including the taxonomy related works. Coming to the next is among the plant vector of pathogens. We have to.
Excuse me, only 5 minutes.
Yes, ma'am. I will complete. So we have to complete. We have to know the entire cycle of the host pathogen and vector relationship. Then only we can identify the vulnerable points so that we can hit the points to get the successful management practices. So far, many varieties are claimed and released as a resistant, but the durability of the resistant is question mark. When we say the resistant variety, when we put it in one place or in another place, that becomes susceptible. So when we put in the even replication tier, some of the accessions are showing, means some of the accessions are less infected in another replicated plot, it shows the highest incidence. So the ranges are very quite high. So we cannot take as a durable resistant lines as far as phyllodes resistant is concerned. So we have to focus more on releasing the durable resistant, then only the sustainability of the sesamum in the farmer's field will take place. So including not only the released varieties, germplasm also we screened lot, but as far as durable resistant lines we did not get it. And attempts were made for screening and also the interspecific hybridization, especially the alatum, malabaricum, mulayanum, radiatum, prostratum, lacinatum, all we used. What we found, because of the ploidy levels are different, the crossability is very difficult because the incompatibility takes place. One thing, even if incompatibility we can overcome by polyploidization, by coleoploidization treatment. But the plant produces the flowers, but it won't sex the fruits, mostly the sterile. That case also we found difficulties. Even few fruits are found, we could not found any seeds which are healthy. It won't germinate, even if germinate the second generation, that two generation star again produces difficult, means it won't sex any fruits. These are the barriers we found. Ultimately, even if few plants we get, and we will do backcross with the donor plant to get that. If the plants goes to the means morphology similar to that of the Sesamum indicum, then the susceptibility associates and comes to the expressions. So as far as the interspecific crosses, we are at the crossroad. We could not find any path breaking incidences in the interspecific crosses hybrids concerned. So one of the attempts were earlier made in TNAU also, that accession also submitted. The similar way we have followed in Sesamum using Sesamum alatum, crossing with Sesamum indicum. But we found these are the scenario found it. Means taffy, or you won't get the seeds, won't germinate, or the plants goes near to the morphology of the Sesamum indicum, that ultimately becomes susceptible. So these are that. Our strength and weakness, we have the huge research team, and also the genetic potentials are very large, and we can use and explore those things. But we have equally constraints. Interspecific crosses won't produce sustainable things, or we could not achieve the tolerable or resistant. We could not generate the resistant lines in successful manner. So these are our strengths and weaknesses. In that, the management approaches we are focusing for cultural and chemical methods, because we have to revolve around the cultural methods and chemical methods. The cultural methods, disease escape mechanism is the prime one. So we have to alter the date of sowing, or we have to use the place where the vector population is very less. And we can introduce some of the intercrops like red crab, so we can arrest the movement of insect vectors, thereby we can reduce the incidences. In the case of chemicals, so any systemic insecticides can be used, but one disadvantage is we have to use frequently to control the jacid population, because jacid population spreads very in the short span. The population increases, and it's also having the quite vast host range. So points to the point are disease is not reported in USA, China, Canada, and Australia. It's surprising. And vectors of the relative sesame infected not yet studied. Only the sesame indicum vectors are studied, and the other species are not yet studied. Some question marks raised when the sea breezes are above 20 to 30 km per hour. How the vector lags, because it's flying insects just 50 meters away from the sea waves. How the insects sits, that viviparous insect sits and feeds and it transfer the phytoplasma. It is a very surprising one. We could not found. And identification of most favorable niches for the sesame to produce phyllodes-free crop, and whether we can develop whether we can go for the genome editing for producing the resistant germ blossoms or germ varieties. So I thank DPD for funding this project, and also ICR and PGR and TNAU as a collaborator in the DBT funded sesame project. So some of the publication also we made. Thank you. Thank you for listening.
Thank you, Dr. Trimaswamy. The forum is open for discussion. 1 or 2 questions.
So, very nice presentation, Trimaswamy.
Thank you, sir.
Very good initiative through the TDBT project, I know that.
We are collaborating with
Yeah.
TNAU,
Yeah.
Raja and other team.
So, the susceptibility factors were identified for this phytoplasma?
Pardon, sir?
Susceptibility factors.
No, not yet.
Not yet.
Not yet.
So, are working on that? Or
No,
how
sir.
you are approaching to that genome editing?
No, sir, only this. We are not focusing on genome
okay.
editing. I think Sendil Raja is working.
Breeding aspects.
Yeah, only breeding aspect. We are doing pre-breeding work.
Okay.
Our institutes does the collection, exploration, evaluation, including the cultivated type and also the wild species. So exchange and also the quarantine. These are our NPPG major activities. Our institute is located, headquarters is in New Delhi. There's National Bureau of Plant Genetic Resources, which is having the 10 regional stations. Our mandates are collection, exploration, collection, evaluation, exchange, and quarantine. These are the major activities that we are doing.
Just want to add a point here.
Sir.
See, very nice presentation, and you gave overview that we don't have any resistant stores. Only Americans have, Australians have. So whether
Not like this, sir. They didn't report the disease, sir.
They
They are not reported the resistance.
Certainly not there.
The disease is not yet reported
Right.
from these
Okay,
countries.
final point, what you mentioned is a CRISPR-Cas technology.
We may approach.
Because there is a dedicated, task force for this. I'm one of the task force members. You propose a project, who is doing it in TNAU?
Sir, Sendil Raja is.
Oh, he is the person, okay. The takeaway from your presentation is he proposed a project, make sure that it is funded under TBT, because there is a dedicated funding only for this. Any disease or any factor which is cannot be managed with the routine process, certainly will be able to help you on that. So in that particular sense, your presentation is very relevant. We'll support that project.
Thank you, sir. Thank you.
Thank you.
Okay, thank you very much for the questions. Maybe now we can move to the next speaker, which
Thank you.
is Luis. Thank you. Please welcome Dr. Arish. He's a professor at the Agricultural College of Dharwad, professor of entomology, and he's going to tell us about constraints about quality peanut production and the biotic challenges facing this production.
Good morning, everybody. Respected chair, I delegate from French and Indian scientists. I am here to deliver my talk on biotic stress, which pose challenges to quality peanut production. So, as you already know, the nutrient value of the peanut, it is very rich in protein and also the minerals, and also it is also has been recognized by UNICEF as a ready-to-use therapeutic food to treat the malnutrition of children in Africa. So these are the major pests and diseases which pose a challenge to the peanut production, but all the diseases are not occurring throughout the country. So some of the diseases are localized, you can see collar rot, stem rot, soil-borne diseases, and even leaf spots, the foliar fungal diseases, and also nematodes. Then you can see the collar rot. It is major disease in northern India, that is mainly Bikaner, Ludhiana. So most of the population, even up to 80% loss is caused only due to collar rot. So initial population, if there is no proper population is maintained, so we can expect huge loss in peanut production. Then stem rot, so it was threatening during 2010. So in Gujarat, Hanumangarh, Latur, so it was severely infecting the varieties. So GG20, it was found to be very susceptible to this disease. Then some of the management strategies they followed by using trichoderma viridae, and also they brought up new resistant strains for this managing this stem rot. So now it is something under control, so farmers can now produce peanuts without this disease. Then leaf spot disease, so leaf spot disease are majorly occurring in South India. You might be knowing Aliyar Nagar in Tamil Nadu, it is an hot spot for this leaf spot disease. Then rust, so peanut above 70 days old, it is prone to rust disease. So some of the states, they treat rust as a maturity indicator, but some of the states it causes severe defoliation, so there will be loss of hull, which is a very nutritive fodder for small ruminants and also the milch animals. So Gujarat, they mainly grow groundnut for the hull. So the livestock in Rajasthan and cattle mainly depend on the fodder of this groundnut, which is very nutritive, and it also helps in the increasing the milk yield. So you might be knowing about the Gir cow and the quality of A2 milk that is produced. So one of the factor for quality milk production is the groundnut hull. So similarly, Fusarium wilt, it was reported by our colleague Tirungalai Swami from SRI of Jodhpur. So it is a localized disease, it is a complex disease caused by Fusarium. So this was reported from parts of Rajasthan like Jodhpur and Bikaner. So sir has devised some of the integrated approach to manage this Fusarium wilt.
Damage like Spodoptera, Helicorpa, and leaf miner. So leaf miner, they are mainly affecting the foliage in Raichur and also near Anandpur area. Similarly, Spodoptera and Helicorpa, it is a polyphagous and it is occurring throughout the country. It also causes severe foliar damage. And hairy caterpillar, it is mainly endemic to Pavgada area, where it causes severe foliage damage and also lead to yield loss. Similarly, thrips and leafhoppers and even aphids, they are common in summer groundnut, where groundnut is grown during February afterwards. Then these white grubs, they are also becoming severe pests due to change in the cropping pattern. So earlier this white grub was not there in Gujarat. When the cotton becomes susceptible to pink bollworm, so the people of Bhavnagar and Amreli and Rajkot district, they shifted from cotton to groundnut. So the area which was near to the forest area, it was started damaged by these root grubs. So initially in the year 2014 and 15, reports were there from Rajkot regarding the infestation of white grub. Then within 2016, you can see that entire Saurashtra district, that is 5 districts which come under Saurashtra like Junagadh, Rajkot, Amreli, Gir Somnath, so all were found to be infested with this root grub. So there was almost 80% infestation. So farmers, they were searching alternate approaches, or they were trying to take take up other crops than groundnut because it completely devastated the crop. So as I already told, Helicoverpa and Spodoptera. So with the proper approaches, we can reduce the yield losses by insect pests up to 48% in case of pods, and also 42% in case of fodder yield. We can reduce by properly managing these insect pests. So as I told, in the year 2016, so it has reached almost 80% damage. So there was a pressure from ministry to take up projects on managing the white grub. So we planned an integrated community approach. So initially we tried to know what is the life cycle. So as already we know the life cycle, it is having a 1 year life cycle, but the challenge was it was having mixed infection. So earlier the Holotrichia serrata, it was confined to South India, and Holotrichia consanguinea, it was infesting groundnuts in North India. But when we surveyed and collected the samples, we could find around 14 species simultaneously occurring in the field. Some of the Adorata species and Anomala species, they were able to complete 2 life cycles, because due to prolonged sowing of groundnut, means after Kharif also, in summer also they have started growing groundnut. Earlier they used to take groundnut during Kharif, and summer they used to shift for sorghum or other crops. But as groundnut become remunerative and they were started to export, they started cultivating groundnut throughout the year. So this has led to the build-up of the population, and mixed infection was also found, was noticed. So we planned a community approach. So these were the strategies, like we have to use the pheromone traps, and we have to use the light traps, and we have to go for seed treatment. So within 2017, you can see up to 85% damage was seen in these 2 adopted villages, that is Samadiyala and Mitapur. So when the farmers adopted these techniques, so we could brought down the damages to 10 to 5%. So now the farmers, they have taken our advisories, and this Mitapur is been adopted by ICR DGR as a model village, and we regularly give demonstration in that village to the farmers. We train them, so they are able to bring down this population, and they are able to produce good, they are able to take good yields. So we are also given them our improved varieties like Girnar 4 and Girnar 5 in this village, which is having rich in oleic acid. This is IOL varieties. So then the peanut industry, that is for export, it is often threatened by storage insect pests. So by damage of this insect, even it adds up to the aflatoxin contamination. So there were several questions during yesterday during Hari Krishna Sudhini's presentation. So this is one of the important pests. So in Gujarat they call it as pupal disease, because you cannot find the infestation unless they come out, and you can see the larvae and pupae. They, after coming out, they pupate in form of cocoon. So observing this cocoons, they caused it as cocoon disease, but this is not a disease. This is caused by the insect Charadon serratus. So this is how the farmers after harvest, they dry the peanut. So it is the first source of infestation. So in field level itself, they start lay eggs, but through eyes we cannot observe the egg laying. So after hatching, they enter into the peanut kernels and they cause damage. So as Sudhini sir said, so first level of defense is seed coat, means pod, then second level of defense is seed coat. So after egg laying, it enters to the pod, and it also infests through the seed coat. So it is an easy entry for the aflatoxin. So we also observed that aflatoxin is more when they mix gleaned pods with the harvested produce. So gleaned pods should not be mixed with the produce. If we avoid mixing gleaned pods, we can also reduce the aflatoxin contamination. So this is how after harvest they some they store bulk storage, they store in gunny bags, they store in fertilizer bags. Then this is the bulk storage method. And this is some of the farmers, they also use tin bins to store the produce from insect pest damage. So one of the innovative farmer in Gujarat.
Excuse me, only 5 minutes left.
Yeah, I will complete. No issue. So you can see the PVC pipes. So entire room he has covered with polythene, this one, and he has stored peanut in a bulk manner, and he has inserted 5 pipes for proper aeration. So leaving 1 feet height from the ground, he has inserted a pipe, so it ensures the circulation of the air. So if the temperature is more, the air escapes through the pipe, and it is also easy for fumigation. They put fumigants through this pipe, so by this they can store up to 1 to 1.5 years without any damage by insects and also aflatoxin contamination. So this was the innovative farmers. We can find so many innovations by local farmers in Gujarat. This is one of the innovation by the farmer. So this is how, when it comes to the market, so it is often neglected in APMCs, and it also becomes the source of infestation by insects and aflatoxins. So this is the common method of fumigation, where they use aluminum phosphide phosphine to fumigate and to manage insect pests. So we also try to know the alternate hosts of this Caryodon serratus apart from groundnut, where it survives. So these are some of the trees which are normally grown around the fields of the groundnut fields in Gujarat, that is cassia, fistula, then albigia, then prosophis. So you can see the egg, it is highly microscopic, and we are not able to see them through naked eyes, and farmer often they neglect, and these hatch and they bore into the pots and cause damage, and also secondary infection by the aflatoxin. Even it has increased this oats range, so it also started infesting rain tree, Semenia saman, and then Acacia nilotica. Then tamarind is the main host. From tamarind it has shifted to groundnut, and it has become major in groundnut pest. So earlier it was reported on tamarind as a tamarind beetle. So now it is started infecting, even in camel foot also it lay eggs. You can see the eggs. So we have done choice and no choice test, and we could able to find that. So the primary preference is given by this insect is to the groundnut, then tamarind, then other host species. If groundnut and tamarind are not there, they are able to lay eggs and also complete the life cycle. So these are some of the tolerant varieties identified through AICRPG groundnut for insects and diseases. This is the list of the diseases and insects, and also the resistant varieties like Girnar 2, Girnar 4, and also KDG 128, and some of the even germplasm lines, and that is CS19 and CS319 resistant to stem rot. So these are being used by the breeding programs, and also they are regularly developing resistant varieties. So I like to conclude by saying that a single method cannot be applied to manage these insect pests. So we have to follow an integrated approach. Like for white grub, it completes entire life cycle, but we have to know the biology of the insect, where we can target and we can have a proper control. Like once it reaches the third instar, it can go even up to 1 meter into the soil. So whatever the chemical you apply on the top of the soil or crop, it will not reach to the pest, and it will be a waste of chemicals and also time of the farmers, and ultimately lead to the yield loss. So we have to manage during the adult stage by using pheromones, by using light traps. So in this tricky is that if a light trap is or a pheromone trap is applied by the farmers, all the pests they are attracted to the single farmer. So here we have to have a community approach. So all the farmers in that area, they have to adopt this technology. So in Mitapur village, we used to go, they were taking tractor sprayers from 7 to 9. They used to spray on the host plants like neem, bear. So they used to place the pheromone traps in the selected trees. So with a distance of 1 kilometer, we used to identify the trees, we used to place the pheromone, and they used to take up the sprays. So by following rigorously, within a 1 year, we could bring down the damage from 85% to 5 to 10%. So if a whole village adopt these kind of technologies, or if they are aware of such integrated manner, so through a team we can reduce the pest population, and also we can contribute in production of quality peanuts. So there are already many speakers have told about integrated approaches like cultural management, pheromone traps, biological control, then forecasting, monitoring. So all these can be integrated for quality peanut production. So I want to acknowledge ICR DGR for supporting me for conducting this work, and also US Dharwad for allowing me to present here, and also TNAU Senthil Raja for inviting me to deliver the talk. Thank you. Thank you, Anandal.
Thank you very much, Dr. Arish, for this very interesting presentation. Is anyone interested in asking questions? Do you have questions in the audience?
Yes, sir. Metarhizium is highly effective, but we have to apply in the initial stage. Otherwise, it will not reach the insect. So, one of the innovation by the farmers of Gujarat is that they used to give urea and irrigate it. So after 1 hour, due to this nitrogen or ammonification, ammonia gas enters to the soil and it makes them grubs to come up. So after 1 hour, they used to spray metarhizium. So it has more chance of coming contact with the metarhizium and kill the this one white grub. But if it has already crossed the third instar, so metarhizium takes little more time to control. By during that time, it used to damage the crop. So we have to have a integrated approach. So in the next year, we have already recommended to mix the metarhizium with organic manure and give it to the soil, and even also applying neem cake. So these approaches are followed. Even NBIR has came up with a formulation that is insecticide resistant MA4 strain, that is Metarhizium anisopli 4 strain. So it is compatible with insecticide. So they tell that you give chlorpyrifos, after 1 day you apply MA4 strain. So after applying, when it comes into contact, the insect become docile. So the more chances of this metarhizium to come into contact with this white grub, and it will give more efficacy in managing this white grub. And some of the nematodes, Stenornima shori, it has been also found effective in managing this white grub. So we have to have an integrated approach to have manage these insect pests.
Yeah, now that the chlorpyrifos is also banned. No?
Yeah.
Not used.
Yeah.
Thank you.
Yeah.
Thank you, Harish. Now I call upon Dr. Terry Marson to present his topic on fungal genetics at the services of wheat breeders. Sir, please stick on 15 minutes only.
All right, so this seminar is a very, very nice opportunity to meet Indian colleagues and to discuss about phytopathology. So I'm very happy to be here, and I would like to thank Dr. Senthil Raja and Professor Hamlin for organizing this seminar. Of course, all the people who also contributed to the organization, especially here in India. So me, I'm Thierry Marcel. I am working at INRAE in France in a unit which is called Biogère, and we are located in University Paris-Saclay, so it's near Paris, not in Paris, it's just very near Paris. And I'm going to talk about fungal genetics, as you are going to see. So Biogère is a unit which is specialized in phytopathology. Everyone in my unit is working on fungal pathogens of main crops in France. So here we have the models on which we are working. So we are working on Leptosphaera maculans, which is causing a disease which is called FOMA on rapeseed. We are working on Coletotrichum ingicianum on bean, but we are also using Arabidopsis. Botrytis cinerea as well, so on grapes, but it has a very large host range, so on different hosts as well. And we are also working on different fungal pathogens of wheat. So we are working on rust, the 3 rusts actually, so Puccinia striiformis, Puccinia triticina, and more recently also Puccinia graminis. And Zymoceptora triticea is also a very big model, and I'm going to talk about this specific fungus. I'm going to be a little bit quick on some slides, trying to gain some time. So on this one, that's just to say that we have very large collections of isolates from these different fungi in the unit. So the inventory, we had almost 40,000 strains in the collection. So that's a very good resource to do research. Also a very good resource in genomics, genomic platform in Biogère. So there is a website where we can find all of our genomic data. And just the example of triticea, so we have different reference genomes for isolates coming from bread wheat, isolates coming from durum wheat as well. We have genome browser so that we can search within these reference genomes. We have no more than 1,000 different isolates that have been resequenced by Illumina, and we have a very new gene annotation of the genome. So I'm going to talk to you about Septoria triticea blotch, which is a disease caused by Zymoceptora triticea on wheat, on bread wheat, on durum wheat actually. So you can see here the symptoms of this disease. So it's on the leaves, and you have some chlorosis on the leaves, some necrosis. And within this necrosis, you are going to see some black dots, which are the pycnidia, which are the sporulating structure of the fungus. So Zymoceptora triticea is an ascomycete fungus. It has two mating type 1 and 2, that needs to encounter to do sexual reproduction. It's a latent necrotroph, so it means that during the first stage of the infection, that you can see here, the fungus is going to germinate on the surface of the leaf, to penetrate into the leaf through the stomata, grow quite slowly into the leaves, and that during at least 10 days there is no symptoms. We think there is no interaction between the plant and the fungus during this time, but suddenly after 10 days you have the expression of the effector of the fungus, and then you start to have the necrotic phase. So where the leaves start dying, the tissue starts dying, and the fungus is growing much faster and starts to sporulate. One of the characteristics of this fungus as well is that it has two modes of reproduction, so it can reproduce sexually and produce ascospores. So that will be mostly at the beginning of the epidemic seasons. It's coming from wheat residues from the past years. So it's really starting the epidemics. And then you have asexual reproduction producing pinidiospores, and that will build up the epidemics during the season of wheat growing. It's because of this dual mode of reproduction, sexual and asexual, when we have a very high level of genetic diversity in the population of the fungus. So that's a paper that we contributed recently, and it was an estimation that in 1 hectare of normally infected wheat field, we can find up to 14 million of different pathogen genotypes. So I'm not talking about spores, I'm talking about different genotypes. So there is really a huge diversity in a population of Zymoceptora triticea. Genetics are quite simple, so it's because it's haploid, small genome of 40 megabases. It has 21 chromosomes, but 8 of these chromosomes are accessory. It means that they can be present or absent, depending on the isolate that you are looking at. More than 13,000 genes have been annotated. And what is nice also is that we can control crosses between different isolates, so in the lab. So that's also very helpful to be able to do genetics afterwards. This paper was also published 2 years ago. We look at the worldwide genetic structure of Zymoceptora triticea, because that's a pathogen that we can find everywhere, basically, where wheat is grown. It's especially strong in temperate regions. Here we used more than 1,000 genomes that have been sequenced of the isolate, and we found that there is 11 genetic clusters which are differentiated, not mostly linked to continents. So you can see here in America we have 2 genetic clusters in North America, 1 or 2 in South America as well. In Europe we have mostly 1 genetic cluster for Zymoceptora triticea, a little bit more of diversity in the south of the Mediterranean and in Asia. And one of the things is that, like my colleague 2 days ago showed, that in Central Asia, and especially in India, we don't know what's happening. And that will be very important to look at what's happening here, because it's just in between those 2 different genetic clusters. So that could also be a subject of collaboration and of discussion together. And what was nice is that we saw that this genetic structure of worldwide population of the fungus seems to retrace the historical spread of the host, so where wheat has been grown and spread its cultivations. So it seems to match very well with what we see in the population of the fungus. Why studying genetics of pathogenicity in this fungus? For many different reasons, because we would like to understand the mechanisms of the infectious process, how this fungus is working, because we think it can help us to enhance durable resistance in crops. So for example, we can have approaches like effector-assisted breeding. Also having knowledge of pathogenicity of this fungus could help in monitoring and predicting the evolution of the pathogen and the evolution of overcoming for resistance genes, for example. In the interaction between wheat and Zymoceptora tritici, so big part of the interaction is based on gene-for-gene relationships between resistance genes in wheat and virulence genes in the pathogen. So it's following the classical pathology model, zigzag model, that says that we have some pathogen effectors which are very important for the pathogen to be able to infect the plant, so to overcome the resistance, the defense. Then the plant may have some resistance genes that recognize these effectors. Then they are becoming avirulence. When there is a recognition, then we have complete resistance, and again, and so on. So we can have new effectors leading to susceptibility, and new resistance genes leading to resistance. So that's why we call it the zigzag model. So in wheat, we know 23 major STB resistance genes already to this disease, and many QTLs which are more leading to quantitative resistance. And one of the questions we had was this gene-for-gene relationship in this pathosystem, is it only for those STB major resistance genes, or can we also find it for quantitative resistance? So for that, we developed a program using genome-wide association studies within the fungus, so to do fungal genetics. We developed already several different panels to do GWAS in Zymoceptora tritici. So 2 panels of isolates coming from France. So the first one here is isolates coming from a little bit everywhere in France, from 2 different cultivars. We had another panel where we have only 2 locations where we collected the isolates, but still from France. And we have 2 other panels, one where we have isolates coming from 8 different countries in Europe and the South Mediterranean as well. So we selected countries with contrasted climatic conditions to do some specific studies, looking at the adaptation of the fungus. And we have one panel that is specially dedicated to durum wheat in Western Europe and to look at pathogenicity on durum wheat. So I'm only going today to talk about the 2 first ones collected in France, and I will start with this one. So it's a panel of 103 isolates that we phenotyped on 12 different cultivars carrying 12 different STB genes, with the objective to identify the avirulence genes corresponding to these STB genes. So, like I said, we have a lot of genetic diversity in the fungus. So here in this panel of 103 isolates, we identified more than 700,000 SNPs, polymorphisms, that we can use for genetics. So that's a huge number of markers. We have a very small linkage disequilibrium decay, so it's less than 500 base pairs. What it means that if we find an association, we are very close from what we are looking for. So it's very straightforward when we find associations to go to the candidate genes in the fungus. And we have no genetic structure because we are at the French level, so that's also very good to do GWAS. We have less bias. So that's the results of this first study. So it's a bit complex figures, but all the dots here are all the associations that we found. Basically, we found 69 candidate genes. We think that 19 of them are especially interesting because they are also differentially expressed during the infection. And what we could conclude from this first study already is that pathogenicity in Zymoceptora tritici is a complex polygenic architecture. There are many genes which are involved. Mostly we have quantitative effects. And all the associations that we found are really specific, or most of them are really specific to the cultivar. So it's very host specific. There are only few, or you can see here, for example, you have several dots on the same lines. It means that this association was found several times. It's for one avirulence gene. It's AVR-STB6 that we identified, that we cloned, that corresponds to the resistance STB6 in wheat. And this AVR-STB6 gene is a small secreted protein, so it's very typical from effectors and from what we could expect from avirulence genes. It has a very strong effect, so when AVR-STB6 is recognized by STB6, we have complete resistance, so it's a qualitative resistance. So it was identified by GWAS, like I said. One picture was missing, but we also found it by QTL mapping. We also identified this one, so the AVR-STB20, corresponding to STB20 in wheat. Similarly, it's a small secreted protein. So again, what we could expect, that was detected by GWAS and also by QTL mapping. The difference of this one is that the interaction of AVR-STB20 with STB20 leads to quantitative reduction in symptoms, so more like to a QTL. So indeed, we can have gene-for-gene interactions in qualitative and in quantitative disease interactions. I'm going to pass on this one. I'm going to talk about the other panels. So this panel, VISA, that was also collected from France. Like I said, we collected the isolates from 4 different cultivars with different level of resistance, and we phenotyped all of these isolates on those 4 cultivars. We found here again, same approach, 11 candidate genes. We are working on several of them. We started with this one. It's a metalloendopeptidase gene that was interesting, identified on chromosome 2 on cultivar Absalon. What was interesting here is that even so we only identified this gene on one specific cultivar, when we are doing knockout of the gene, so in different isolates, when we are doing from wild type to knockout, we increase the level of aggressiveness. So when we remove the gene, the fungus is more aggressive, and that's independent on the cultivar. We found it on this one and also on this cultivar. So this one is not working in a gene-for-gene relationship. What was interesting here is that we also phenotyped all of these isolates for fungicide resistance to 4 DMIs and 2 SDHI fungicides, with the idea to look to whether we have some correlations in between aggressiveness of the fungus and resistance to fungicides. And that's the correlation matrix that we have. And what you can see in red is when we have significant positive correlations. So we found it in between the cultivars for aggressiveness on the different cultivars. So makes sense. Even so, we don't identify the same genes, so there is still a common basis. We do identify positive significant correlation in between the different fungicides for resistance to the different fungicides. That also makes sense. But what was the most interesting is that here we did identify some also weak but significant negative correlation between fungicide resistance and aggressiveness on some specific hosts. And that was really specific of the combination between one fungicide and one cultivar. And that could be interesting because if you have that kind of relationship, it means that if the fungus is more aggressive, then it will be more difficult for him to adapt to the fungicide. So if we have some kind of couple between one cultivar and one fungicide with this kind of negative correlation, that can be used also to trying to reduce the adaptation of the pathogen. We did a special GWAS trying to identify the genes that were explaining this negative correlation. So it's a GWAS that takes multivariate phenotype into account. Doing this GWAS, so it's called meta-CCA algorithm. Doing this GWAS, we did identify some small secreted proteins and genes involved in resistance to fungicide that we knew for single traits. But we did identify 2 associations explaining this correlation between fungicides and host resistance, aggressiveness. And one of the candidate genes that we found was the phosphodiesterase, and that was very interesting because we know that in fungi, phosphodiesterase have already been implicated in virulence and also in fungicide resistance, not in the same case, but independently in the 2 cases. So that's indeed a very good candidate to explain this negative correlation that we observed. And so fast, so we did also knockout of the gene in different isolate background, and we found that when we knock out this gene, we do reduce the aggressiveness. And phenotyping for resistance to fungicide is ongoing, but at least it's a very interesting and good candidate. So messages from what I've been saying is that we have a very complex polygenic architecture, mostly quantitative, of pathogenicity in this fungus. We have to have many gene-for-gene interactions which are involved, and that can like qualitative but also quantitative interactions. But we also have, we think, standing genetic variation for pathogenicity genes, which are not specific to the cultivar. So the fungus really has, we think, has the capacity to increase its level of aggressiveness to different weed varieties, independently of the weed variety. And we have these phosphodiesterase genes that have an antagonistic pleiotropic effect that could be very interesting if we think at trade-offs on trying to reduce adaptation of the fungus. What we are doing now is trying to identify and validate new virulence and pathogenicity genes from what we have been identifying. We are also now constructing a pangenome graph, trying to improve our genetics approach, trying to improve our detection of genes. And the idea also of all of that is to use all of this knowledge to have high throughput tools to follow the evolution of the the genes, the pathogens, the virulence in fungal populations. We have 2 more slides, and I want to talk about it. The question was, what is the virulence profile when we look at Zamocetua tritici populations in naturally infected wheat fields? So the 2 last slides I want to show is that the tool we developed as a multiplexed amplicon sequencing to be able to follow the genes that we identified. So for that, that's the process that we use. We use FreeDime. So FreeDime is an array when you can do high throughput multiplex PCRs. And what's interesting here is that you can mix. So we mixed actually almost 800 primer pairs, so targeting 800 targets in the fungus. So of course our targets are all the genes for pathogenicity, also genes for resistance to fungicides that we know. And we have 691 amplicons which are neutral markers. And here it's just to do population genetics in the same time. And on the other side, we can have 192 samples. On each sample, DNA samples. On each sample is not one isolate here. So what we did is that for each sample, so we are using a network in France that is following fields infected by septoria everywhere in France. On one sample is 50 leaves with lesions collected in one field that we grind all together. We extract the DNA. So in one sample, we have DNA from the wheat, but we also have DNA from the fungal population that was present in that field. And then by doing so that multiplex amplification and sequencing by Illumina, so we can get all the variants for each of the target amplicons that we are looking at. So that's the example for 192 samples from this network in France with a neutral marker. So we look for genetic differentiation, but we did not find it, because at this level, French level, we have no genetic differentiation of the fungus. But if we look for specific targets, so here, for example, we look at AVR STB6. So you see that we found 3 different variants all over France. One clearly associated with resistance, so a virulence recognized by the plant, and 2 variants associated with virulence. And then we can map these different variants on the map of France, where we took our samples. And you can see that we have an increase, or we have a higher frequency of avirulence in the south of France, and a higher frequency of virulent isolates in the north of France. So that's really the tools that we want to develop and continue to use to feed also with our genetic studies. And I will stop here, thanking all the funding agency. So my team is the team ADEP, Adaptive and Epidemiological Processes in wheat fungal pathogen interactions, so in Biogère, and all my collaborators in France and also in Switzerland and Spain. And thank you for your attention.
Thank you, Terry Marcel. Discussion, small discussion, one or 2 questions.
Excellent presentation.
Thank you.
I have one, it's not only to you, wherever I listen to such a presentation, I ask this question. How fast the effectors are evolving so as to break the resistance? What's the timeline for the new effector, a new resistant gene getting lost its function?
Well, it's difficult to give a timeline because it will depend on the pathosystem, it will depend on the gene as well. So how fast are new resistance coming, that depends on breeders.
Yeah, from the context of
and that's interesting. Yeah, so that's interesting and very important to work with wheat breeders as well, to know the new varieties and when new resistance genes are being released, so that we can also follow them. And we're actually working on a little bit faster way of trying to identify avirulence genes towards the new resistance genes in the varieties. Because that was all nice, but I think one of the key points after is how fast can we be in getting the information to be able to follow what's happening in the fields. After in Zamocetua tritici, it's interesting because you have most of the time you don't have breakdown of resistance, so it's not from zero from black to white, so it's more like an erosion. Because you have so much diversity in the populations, also because of the sexual reproduction is so common, so avirulence genes remain in the populations. So it's not so fast. Even some resistance genes that have been completely overcome, avirulence genes still exist in the population of the pathogen. So I think for this pathogen, it's especially interesting to see, to be able to follow actually these virulences and to have a little bit of timeline for a gene.
Right, okay. Yeah.
Okay, thank you very
Thanks.
much, Thierry. And let's. Well, yeah, I'm not sure we have time. Do we have time?
Congratulations, it's a good presentation.
Thank you.
I'd like to know what are the other countries where the septoria is the major problem in wheat.
Yes. So sorry.
This sectorial leaf spot is a problem in other countries well?
If the disease is
Globally,
developing,
yeah.
I think the disease is about everywhere. Yes, you can find it. I mean, you saw the worldwide study that was done, and you can really find Zymoseptoria everywhere. After the incidence, it is very different from one country to the other, but also from one year to the other. So if you take like the 3 last years in France, for example, the last year we almost didn't have disease. I mean, all of the experiments that we did this year, we don't have disease developing at all, just because the climatic conditions were not good enough. But 2 years ago it was a drama. That is, this disease was explaining most of the difference in yield that we could observe in France. So I think you find it everywhere, but climatic conditions are very important, and from one year to the other, the incidence might be very different.
What is the yield penalty, yield loss due to this disease in your country?
I don't know, if you have a susceptible variety, it can go over 50%, but we don't have so much with susceptible varieties anymore. I think that was a big target for wheat breeders in France, but I don't know, last 2 years ago, maybe it was 30%.
Okay. Any other major disease in your country?
Any other disease? Yes, of
Any
course.
Major disease in wheat?
Yes, of course, there are many other diseases in wheat, yes. So I think that one is especially important. In Europe, it's a disease that is causing most of the fungicide sprays. So that's why also it's very important to find solutions, other solutions than fungicides to fight against this disease. But rusts are also very important in France.
Okay, thank you very much, Thierry. And let's join me to give Thierry a round of applause. So now it's my pleasure to welcome Professor Parani Dharan. He's a professor in the Department of Plant Pathology here at TNAU, and he's going to tell us about mycotoxins contaminations in food and animal feeds in India.
Yeah, good morning to everyone. Really, I'm thankful for the organizers to giving an opportunity to present my paper. So today we are going to discuss about what is the current status of major mycotoxin contamination in food and animal feeds in India. Actually there are recent problem occurring in both rice and permanent problem in maize also. So ochratoxin is one of the main toxin in rice in addition to the aflatoxins. So we know very well China and India are major exporters. The major consumption per person is almost 57 per kg of rice. The consumption is carried out by human beings. So the ochratoxin is produced by Aspergillus ochraceus and Niger, and penicillium also capable of producing these toxins. So it's a kind of carcinogen, it's capable of producing cancer. So that's why FSI and the European Union also are having a lot of restrictions to both import and export. So for the purpose, unprocessed cereals only is allowable limit is 5 microgram per kg. And if you go to baby foods and infant like foods, so only is a 0.5 microgram per kg. So based on this report, we have carried out what all the microbiota which is present in the normal polished rice in Tamil Nadu. So we have isolated lot of microorganisms from rice, but other fungi is not a problem. If you wash it, we can eliminate it. But this Aspergillus, especially ochraceus, is a problem of producing the toxin. This toxin go and imbibed into the starch area or inside the polished rice. So it's permanently present. So for that purpose, we have quantified what is the real toxin which is present in the rice and all these things. So before that, we have isolated some of the isolates which was isolated from the normal rice which is available in the markets. So we have isolated and we have quantified what is a virulent strain also. Just is a basic study we have tried. In addition to that, what is the real temperature and what is the moisture which is needed to produce the toxin. So this is the basic data we needed. For that purpose, we put a different temperature and water activity. Then we measured what is the growth rate of Aspergillus ocraceus. So already yesterday also, research scientists cleared about some other things. So even 25 degree is enough to produce more amount of toxin, and also growth rate also very high starting from 25 degree. So from 25, 30, 35, its growth rate is not affected. So and also the kind of degree temperature is also even 25 degree is enough to induce more amount of toxin in addition to the water activity. So it is actually xerophilic fungi. It's even it's capable of surviving in the dryness. But if you give the moisture, it's automatically it's aggravated. It's capable of producing more toxin. So for that purpose, we have analyzed the rice material which is available in the market. Even the household also we have collected. We have tested what is the ochratoxin which is present in the samples. So if you see this, some of the samples are exceeded the European limit. So European limit only 3 microgram. So India is allowing almost 5 microgram, but European and Japanese peoples are expecting the 0 microgram. That means nothing. It should be free from toxin. So like that, above the permissible limit, European Union also some of the samples are, we have detected more than the 3 microgram per kg of ochratoxin. So previous one is actually it is a brown rice. So some of the hilly region in Tamil Nadu peoples are using the brown rice. So we have tested the both brown rice and polished rice. So polished rice also we have detected some of the samples are having the above the permissible limit of ochratoxin. So already I told this it's a kind of ochratoxigenic, sorry, it's a carcinogenic in nature, capable of producing the cancer. So these are the some of the chromatographic things we have detected. So this is the top one is the brown rice, next is the polished rice, and some of the standard also we have compared with the free samples of ochratoxin. So this is the total number of samples we have collected, what is exceeded the limit like that. So almost 12% of the samples we have collected with this around 50 samples. So 12% is exceeded the European limit. So we have to be in the very dangerous zone to export or import a kind of rice material. So for that purpose we have screened some of the biocontrol agent which is capable of having the antagonistic nature to prevent the growth of the Aspergillus ocraceus and also able to degrade the toxin. So we have screened lot of baseless species. So out of different antagonistic nature, this baseless amylo liquefations are capable of both growth inhibition and also it's capable of reducing the toxin production. So these are the some photography of the inhibition zone, which is producing the baseless amylo liquefations and valencies like that. So when if you go for the inhibitory effect of the, we have tested the volatiles which is produced by this baseless amylo liquefations. This volatile is almost 67% is degrading the ochratoxin, just volatile compound which is present in the amylo liquefations. So for that purpose we have screened with the GCMS analysis, we did the metabolomics. So identify the sum of the compounds, these are the sum of the compounds which is maybe the responsible for the growth inhibition and also the detoxification nature. So this is the first case of ochratoxin. Next, the recent disease in India and South Asian countries is the rice fall smut, but it's not a original smut, belongs to the classification. That's why it's a rice fall smut. So it's produced by acetylauxin toxin. So it's almost affected all over, spread all over the world. So it's capable of producing the very important toxin is called as acetylauxin. So this acetylauxin also having the capable of some of the carcinogenic in nature. So actually the symptom is just it's the grain is transformed is a mass of spore balls. So you can see these yellow spore ball or orange color spore balls are converted. So it's later it produces the chlamydospores. So it's spread all over the world. So these toxins are released when is a highly hydrophilic in nature, easily disposed in rain splash, and also most of the rice are in aquatic condition. So semi-aquatic condition water also contaminated with toxins. So it's accumulated and translocated to the non-infected part also. So it's a fall on the water. So it's a hydrophilic nature dissolved in the aquatic water. So it water also it's absorbed by the healthy plant. So automatically healthy plants also affected by this toxin. So these are the several problems in the rice ecosystem slowly developed because of this toxin. So actually it's capable of producing a 3 type of toxin: acetylauxin, acetylorganidins and sorbinolidins. These 3 toxins. Out of these 3 toxins, acetylauxin is a major problem. It's having the negative impact on the eukaryotic organisms. It's having the tetrapeptide structure. So that is a main toxin content. In addition to the things, there are 6 type of toxins also that is present within the acetylauxin, that is A, B, C, D, G, like that. So what is the impact of acetylauxin? So acetylauxin is preventing the cell division by the inhibiting the mitosis. So during mitosis it's preventing the microtubule is very important for separation of chromosomes during the mitosis. So it's capable of preventing this phase. So it's bind to the tubulin protein and preventing the microtubule formations. So in karyote show the anti-mitotic activity also. It's a highly lethal to the eukaryotic organisms. So we have isolated some of the Astronogeroide virus fungus. We have tested the toxins. So problem is it's a kind of a new toxins. There is no standard is available in anywhere else. So we have to purify the toxin. So then only we can compare the any kind of experiment. So for that purpose, we have tried with the toxin extraction with the ethanol and water fractions with the LCMS and MALDI-TAFs. We have tried, and finally we have did also the several type of fraction. We have able to identify the acetylauxin D and A like that. So after extracting, now we are testing. So what is the detection of acetylauxin which is present in the aquatic, semi-aquatic condition of the water of the rice? That is very important because it's having the preventing the mitotic activity and problem in the lethal activity of the rice plant. So for that purpose, we have collected lot of surface water of the rice samples from different field from Tamil Nadu. So we have tested the acetylauxin level in the surface water of the paddy field. So we have detected some of the samples are having the capable of acetylauxin. Okay. So based on this, we have tested the acetylauxin also. It's having the phytotoxicity effect, and it occurred in the different paddy straw and other kind of things. So third part is the maize poultry feeds. We have tested the poultry feeds because Tamil Nadu is the major hub of the poultry area. So for that purpose, we have collected different feed from the different industry, poultry industry all over Tamil Nadu, and we have tested the aflatoxin content. In the aflatoxin content, DDGS and some of the major poultry feeds are contaminated by the these kind of aflatoxin. So DDGS nothing but the ethanol byproduct. So India nowadays is giving permission to produce the ethanol production in maize, but the byproduct is a DDGS is a highly extraordinary infected by the aflatoxin. So we cannot use those byproducts. So these are the some of the HPLC. So we have tested the same way water activity and some of the temperature also, some of the genes also, what are the genes responsible for this aflatoxin production also we have tested. So coming to the mitigation strategies, we have collected the different kind of microorganism Western Ghats. So because Western Ghats is after the Amazon forest, it is one of the UNESCO approved forest. They are having the unexploited microbes which is present in the deep forest. So we have collected lot of soil yeast from Western Ghats. In addition to the some of the native soil also we have collected. We have screened a lot of organisms. So finally end up with some of the bacillus and Saccharomyces having the capable of inhibition of growth growth and detoxification. So we found 2 botanicals are also capable of preventing the toxin growth also. So we got tested all these antagonistic to preventing the growth and detoxification, and volatile also we got tested how the fungus is inhibited by this volatile nature like that. And also we have tested in the HB also. So volatile exposed from this bacillus and yeast, which is having the preventing the growth and inhibiting the detoxification levels. So botanicals are also we have collected and we have tested against this aflatoxin degradation. We found that if you see this is the untreated control aflatoxin production. After treating this botanical, the area is highly reduced. So this is the tested, if the botanicals are mixed with the brain stem, that is a cytotoxicity studies. We have bought some of the basic Artemisia salina brain stems. So this is a controlled one, which is not added by any botanicals, but we have added the toxin plus botanicals. So these botanicals are giving neutralize the toxin. So this here the shrimps are very active, but here only the toxin is added here. So most of the shrimps were died. So these botanicals are capable of preventing the or detoxify the toxin. So it's a living and actively playing here. Okay. So based on that, we are going to take this next level. So based on this, what is the take home message means this ochratoxin, nasotoxin is the emerging mycotoxin, threats to rice ecosystem, because we are most of the world peoples are staple food is a rice. So we have to concentrate on the rice, and also poultry feeds also. Nowadays the children are very fond of chicken, so every country is producing a lot of poultry. So all poultry feeds are affected by these toxins. So we have to take policy decision to curtail all these things. Nowadays these poultry industries are using the clay material to detoxify the toxin, but it's not approved by the World Health Organization or FAO. So we need of a good probiotic yeast or bacillus to prevent the growth of the toxin. So mycotoxin risk depends on temperature and moisture also. So aflatoxin from rice falsement is toxic and water soluble nature and persists in the ecosystems. So we have to need for the integrated management, and eco-friendly degradation is required, and strong monitoring system is required to protect the food and feed value chain. Thank you very much. So I acknowledge the National Medicinal Plants Board. So with this project only we have invented that yeast and bacteria. So thanks to everyone.
Thank you very much, Professor. Are there any questions in the audience? Yeah, please.
You know, we have heard of peanut aflatoxin very much, but the toxin which you are referring in rice, it is least known, or
yeah, it is a listed one. Ochratoxin is also, when you want to export rice, nowadays they are started testing ochratoxin.
Right. And unlike peanuts, rice is cooked, no? During the time all the toxin will be gone?
So sometime we are using owl. So without cooking, but we are using.
It's a small fraction of
So smaller fraction become problem. It's also hydrophilic in nature. It's tolerable to high temperature also. Okay. So we need some basic data. Are missing still.
No. Is there any data indicating our export of rice is stopped because of the acrotoxin?
So far they are not insisted so far, sir.
Okay. So it's emerging.
Emerging.
Another interesting point which I noted is the herbal. Some of the botanicals has deterrents against this toxin. Yes. Which herbal you are referring to?
So the two.
No, it's confidence.
Yeah. We are waiting for the. Okay. That's why I made it to
that
botanical.
Would be very interesting.
Botanical 1 and botanical 2.
Okay. But you will not reveal the name of the plant. Okay, we will find out. Okay.
Yes.
Thank you. Very good presentation.
Thank you.
In fact, those things are least known. In that case, we have to take it forward. Yes. Because we are the major exporter of rice, particularly basmati.
Yes.
I don't know whether you targeted basmati. Basmati has lots of volatiles.
Yes, sir.
right?
Yes, sir.
There are lots of volatiles which deter pathogens.
Yes.
I don't know, you look at it.
That false smut is a problem in all the varieties, sir. All the varieties.
Okay.
Yeah.
Kindly look at it carefully.
Okay, sir. Thank you,
Thank
sir.
you, all the best.
Now I call upon
Thank
Dr. Nalathambi. Thank you, sir. Now I call upon Dr. P. Nalathambi, ICAR, Nilgiris, to present on host resistance mechanisms to combat major diseases epidemics in Indian wheat.
Good morning. Thank you for this wonderful opportunity. Today I'll share the host resistant mechanisms to combat the major disease in the wheat. There are so many diseases, but the diseases which are major in Indian context. As we know that the wheat is fulfilling about 33% of the food grain production in the country. It is the second major cereal crop, and but still we need 125 million tons to meet up to the population in the year 2050, which will reach about 1.7 billion. Since 1965, the country has released about 545 commercial varieties in the country. Still, these varieties are under cultivation, but still it is estimated that about 20 to 30% loss, the overall loss, could be the dominance of herbicide resistant weeds. It is the upcoming problem in the northwestern part of the country. And of course, the imbalanced nutrition obligation by the farmers and inconsistent irrigation, because the water availability is the problem in the northwestern part, and particularly in the desert areas like Rajasthan, Gujarat, and of course the lack of terminal heat resistant varieties, because the wheat is grown in the Rabi season in the North India. When it approaches the maturity, the heat will be more. So we don't have very tolerant resistant varieties which can tolerate the terminal heat. So these are the major factors which may contribute about 30% loss. But still, the epidemic disease, it's uncertain, and it will affect the productivity and quality every year, either one or other disease. So as on priority, earlier the stem and leaf rust were in the top, but now yellow rust or striped rust is taking upper hand, and followed by the powder mildew. It was the minor disease, now it is the major disease. And of course the head scab, Fusarium head scab. Just now my friend discussed of the toxin, effect of toxin. This is another dangerous fungi. It is significantly affecting our export potential. And of course kernel bunt, everybody knows, it's a quarantine disease of the country. We can't export. Some countries are having the zero tolerance. And the leaf and stem rust, it is always there. Coming to the wheat rust in the Indian varieties. So before our green revolution, the rust epidemics resulted heavy yield loss in the country. Therefore, we used to import the wheat grains from the other countries. But our pioneers like Dr. K.C. Mehta and other group, they worked very systematically, and from 1965-67 onwards, we released our own varieties which are resistant to the rust. So these are some of the classical varieties in those days, 60s and 70s were released, and you can see the classical varieties like Kalyan Sona, Sonalika, and all that were very large scale it cultivated in the northern belt, and therefore we achieved the green revolution. So since then we are fulfilling the requirement of the country. And coming to the rust resistant genes, our own varieties, because we very nicely picked up the hypothesis of the gene for genes hypothesis from the flower, and this is the one of the wheat is the best crop, the best model in the country where the host resistance is still playing the major contribution for the food production. So the origins of the host and the effectors of the pathogen, it is always fighting each other, but still we are able to manage with this three category of the genes, that is SR genes, LR genes, and OR genes. These genes are present in the wheat, our own Indian wheat, and we are able to contain these major three rust pathogens. Till date, as on date, we have 47 host resistant genes in the country, our Indian varieties, that 500 plus varieties have this total of 47 host genes. I think my friends, you can explore our the already released commercial varieties for your further improvement, because already it is released with this host resin genes. And these are the genes, 24 SR genes for the stem rust, 19 LR genes for the leaf rust, and 6 OR genes for the yellow rust. Interestingly, some of the genes like LR26, in combination with the other LR genes, has sustained the wheat production till date in the country. Last 4 decades we are sustaining with only with these few genes in our almost all the varieties. Similarly, SR31, all you know the UG99, the India was, they were very apprehensive that Indian wheat production will go down because of the UG99. But we stood very strongly because all our varieties invariably were intercrossed with the SR31. This confer the UG99 trace. Therefore, with this SR31, India stand top in the world. We are not allowing the UG90 in our country. Otherwise, the rest of the Asian countries, other countries, they are having the UG99. So, however, as we understand as a pathologist, the pathogen is very clever. It is always produce its own effectors to break down the resistant genes. So, there are a lot of effectors, and of course, but the host itself, the effector triggered immunity, it will contain the pathogens. So, this fight will be always going on, and in our country we are working very systematically. And of course, the wheat rust pathotypes work started somewhere in 1922, but it was documented somewhere in 1967 through the wheat disease trap nurses in the country. I told that this is one of the best model crop. 60 itself, the country has followed this procedure. Everywhere they put the trap nursery, and they throughout the country wherever the hotspot is there. Then we used to monitor. Then later on we extended this facilities as a SARC nursery. We include the SARC countries also, and even Wellington we are having the SARC nursery, and we evaluate and we categorize, and ascend that we have 145 pathotypes. We are maintaining at our national repository at Florida Shimla. Throughout year they are maintaining under minus 180. Anytime anybody wants the race, we can take, we can test, but we cannot bring the race from one zone to another zone. It is restricted in the country, because otherwise it will, the epidemics will happen. These are the some of the predominant race, and we are strategically deploying the R genes, because I told there are so many genes according to the necessity of the race. Particularly, suppose if I want a developed variety in the southern gill zone, I'll take the race which are resistant to dominant in the southern seals. There are some report about the physiological resistance since we moved ahead with the molecular genetics. So we are a little concerned with this, but still some of the genes like endochitinase, glucanase, and other genes are also used for the identification of the resistance. The another second most important is what I told, the powdery mildew. It's a very tricky pathogen, all we know. It's very difficult to culture, very difficult to maintain. But still, with the support of the funding from the Department of Biotechnology, at Wellington we have worked out some procedures to identify the resistance. This is about the pathogen. And because the main concern is that when we are scoring or screening the varieties for the rust, the powdery mildew will dominate. The actual true type of resistance will be escape. So here we have the powdery mildew disease escape. To avoid that, we need some mechanism to evaluate the host for the powdery mildew first. So at Wellington, and we standardized some of the methods, and interestingly about 275 the collections, the powdery mildew collections we gathered from 3 agroclimatic zones. One is from the northwestern part, another is northern gill zone from Shimla, and southern gill zone, of course Wellington. And we maintained, I think pathologists may surprise how you maintain the powdery mildew. So, but we maintain, and through 2 types of the resistance mechanism. One is seedling resistance and the other product resistance. So here we nicely we use the seedling resistance mechanism to maintain the pathogens. And of course these are the some of the our own Indian varieties which have been identified as a field resistance. So whenever we need, we use these field resistant varieties to counter check the resistance pattern. So this I told, we imported about 35 the isogenic lines from CIMIT, which were conferred with the PM genes, resistant genes. Because in India there was no studies to characterize the resistant genes from the wheat, like in case of rust. So we imported these known genes, and first we worked out the differential reaction against our own Indian isolates. Then we picked up the most virulent one. This is the phylogenetic studies. My friend Dr. Kumar supported this program very nicely. So without support, I think this might not have happened. So with a lot of effort, with consistent coordination, we could bring out, and we identified one very specific virulent pathotype from Wellington out of this. And you can see how we multiplied. A simple sterile agar medium, the first petri plate you can see, it is as good as like any other fungal culture. But here we use the leaf, completely sterile leaf, and we inoculate the powder mildew. You can see it is colonizing on the leaf, supported by the agar medium. And some test tube method also we tried. Then the DBT funded for a separate structure for this pathogen maintenance. So this is design, my own design. I put the structure inside, we put some micro cabins inside the polyhouse structure with controlled condition. This the large number of the seedlings we used to raise in the structure, we inoculate, and you can see the seedling reaction. So then with this we identified the genetic diversity of the powder mildew pathogen. Because I told 275 isolates we maintain, and we utilize the sealing technique to find out the genetic diversity among the pathogen, because that is very important than the host. And you can see the gene efficacy at the PM genes, that is powder mildew resistant genes, efficacy we worked out. So which genes is more consistently resistant against all the pathotypes. So we have seen the gene efficacy. Then we compared this, our genomic data, with other countries. It is already available in the NCBA database, like Japan, France, Austria, German, and our own Indian isolates. We are happy to say that the Indian genetic diversity database is maximum in the world as on date. We submitted maximum sequence, and from there I told that we picked up the most virulent, we studied the variability, then we went for the complete genome sequence. It was very tough task. You can see the single colony, single spore colony, we multiplied the seedling, then we augmented, we checked the quantity of DNA. It took more than 1 year to check the quality of DNA itself. Then we went for the whole genome sequencing. I think in the country this is the maximum, the big genome submitted so far among all the pathogen. And the first powder mildew pathogen submitted the best assembly, hybrid assembly, first assembly of the country. So from this isolate, this is the submission, and now we are multiplying this specific strain or the pathotype. We are supplying this inoculum, because nobody can supply you the powder mildew inoculum. But Wellington can supply. We are supplying to other partners, and they are evaluating. This another disease, Fusarium red blight. I think it's also in the France, your country, the ear blight. I think you are calling it ear blight. So it's another major disease. It is emerging. These are some of the toxin, that is dioxin is a major concern for the export. And the another disease, the kernel plant, it's I think already existing disease. Some of the varieties we have the resistant mechanisms. Now the point I want to discuss, because yesterday I told that I need a few minutes to discuss about the collaboration between your partners, France. That in India we have the excellent facilities for identification and validation of the durability of the resistance. We have excellent facilities at Wellington, and we have excellent facilities at our headquarters Delhi. You can capitalize it for your disease like rust, or the powder mildew, head scab, leaf blight. Of course, if it is kernel plant, since you have the zero tolerance, you can exploit our facilities. I think with this you can collaborate. Let us discuss for the plenary session for the collaboration of the new programs. So with this, let me thank you all. Thank you so much.
Thank you so much, sir. Quick questions from audience, please.
Yeah. Wheat blast, I can't comment. It's a government instruction. I can't comment.
Is it there or not,
Yes.
sir?
No, I can't comment. I told you I can't comment. Yes, yes, because I tell you, because you know that we are already having the problem of the export in the problem of the kernel bund. We have seen zero tolerance from some other countries. Now we are facing problem with the fusarium head blight. If I encourage another disease, then it will be a problem for us. So some scientists published some papers. There are some strict, stringent guidelines from the council that without consent of the council, we should not discuss about this issue. That's why I'm keeping quiet. Otherwise, I can discuss more. Yeah.
It was spreading from Bangladesh in 2016 February, but it was contained very effectively by all kind of regulatory measures. They never allowed the pathogen to cross into Indian territory. That was a government stand, and we declared that India is free from wheat blast outbreak.
Despite that, some Indian scientists published the research paper.
Yeah, that is a status of the state of government. They very clearly told it never created any epidemics in any part of our country, and it was very effectively contained right in the border itself. So India says that our country is not having wheat blast outbreak at all.
After that stand, some stands.
No, it is like that only, and it is true also, because you see 2016 February onwards, even up to today, there is no any outbreak from any part of the country. It was very clear, right?
I think, Dr. Karthi, sometimes scientists are very curious to publish our findings now. So without ascertaining the actual situation of the government, we are publishing in a very hurried manner. It will create the international problem.
Subsequent to that,
that's why.
Even after 2016, a government launched a scheme to monitor the disease outbreak. From that scheme also, they clearly confirmed that there was no any outbreak in any part of the border states and other part of the country. So that is a stand. So I have one small question to Dr. Nalathambi.
Yes.
Any rust-resistant genes, I think it belongs to NLR category, whether it has got any effect on powdery mildew also?
Yes, there are some linked
sort
genes.
I don't know, because we know that our genes are very specific.
Yes.
Why I'm forced to ask this question, because you also encounter both the pathogen together on the same leaf. So is there any co-evolution of some resistance?
Yeah, actually,
for the pathogens?
Yes, for the information, our Indian scientists also published some of the papers related to the PM genes resistance Indian varieties or have some PM genes. The PM genes are actually not characterized, but the PM genes are linked to other SR genes, LR genes. So LR26 is linked to PM6. So when you characterize the LR26, you are presumed that it has the PM6 genes, but it's not the true case. That's why I used to fight with the breeders. First you characterize the actual gene. If you claim the PM genes, first you characterize the PM genes, not your linked genes. Because the linked genes expression may differ at any time, but we also to focus on the major genes.
Yeah, because these two pathogens are coexisting in the same locality or the same genotype. So maybe the long evolutionary history, there are chances that our genes might evolve parallelly to target both the pathogens simultaneously.
Yeah, I think
it's a theory, but we are not sure whether it is really working in the open field.
We are actually, because when we were focusing for the green revolution, we were focusing on the rust only. Is it? So we ignored the most of the other upcoming disease. It will take time. Okay.
Thank you, Doctor.
Okay. Thank you so much.
Okay. Sir, we have a question, please.
Thank you for this very nice overview. However, I was wondering if India is free of any root disease on wheat. You didn't mention tecal, sharp eye spot, for instance. Sorry?
No tecal in India.
Okay, very good. What about sharp eye spot?
No, it's also not there.
Sharp eye spot is rhizoctonia. You know,
rhizoctonia is there. Seedling rot we have. But it's not a major concern because
okay, so it's not
these are all minor disease.
So it's not because it's easier to have a look on foliar and stem diseases than above ground diseases?
Because we won't face, because the cultivation pattern itself in India, if you see the larger area cultivation, before the take up the sowing, wheat sowing, they will go for the opening the field and keep it for some time as a soil solarization. By the time it will take care of all the soil-borne pathogen and insect pest. Therefore, I think the wheat is the only crop where it attracts very, very minimum pesticide or fungicides. In my point, except for the yellow rust, we never spray in the wheat. We never spray any chemicals. So that way, because the cultivation pattern itself is quite interesting in India, particularly in the northern states.
Okay, thank you. That's interesting. Thank you.
Thank you, Doctor. Now I call upon Dr. Sebastian Kunak from IRD, France. He is talking about leaf pathobiome profiling with nanopore. Please, sir.
Good morning, everyone. So first, I wanted to thank the organizers for giving me this opportunity to present our work and in such a very friendly meeting. So today I'm going to tell you about a small project, an exploratory project that got funding from Agropolis Foundation in Montpellier. And so despite the very fancy title, you will see that it's just a prototype, and I'm hoping that maybe in the future it could become more widely used for disease diagnostic and also for other purposes I will try to illustrate. So these are the people that actually did a lot of the work, and especially these two students. So it's not working. Okay. So I will skip this slide. So yes, I don't think now it's necessary to just remind you about metagenomics and microbiota and microbes, because Professor Kumar on the first day gave a very nice broad introduction to it. I just wanted to point your attention to the applications of metagenomics in the field of disease management and plant health monitoring. So you can have surveillance. This is a very important. This could be very important for metagenomics for identification of new invasive pathogens without any a priori. It's also important to try to identify routes of transmissions of pathogens, and also for tracking pathogens and non-harmful microorganisms. Today, the point I would like to make is that it's also potentially very interesting to help deploy effective resistance using pathogen-informed approaches. So the pathosystems we are working on is on rice, and I guess everybody knows about bacterial leaf blight, especially in India, where I was told it's a very serious disease. So this bacterial leaf blight disease is caused by bacteria of the Xanthomonas oryzae species, and it's a special pathovar. It's a pathovar oryzae, so I will call it XOO for short. And this disease is very important worldwide, and it can cause very significant losses. Within this species, there is another group of bacteria, which is another pathovar, which is called Xanthomonas oryzae pathovar oryzicola, or XOC for short, and it causes another disease, BLS, bacterial leaf streak. For the untrained eye, the symptoms may look similar, although they are actually quite different depending on the disease stage. So the impetus for the project was kind of several observations. When we went on surveys in the field, we could get leaf samples with symptoms of what seemed like both diseases at the same time. So this suggested that you can have on one leaf both pathovars, and subsequently we also could confirm using molecular approaches that this was indeed the case. We had in the same leaf co-infections between the two pathovars, and also that within one leaf you could also have different strains from the same pathovar. So these co-infections, for us, it was very intriguing. Maybe we were a bit naive, or I was a bit naive, Okay. I was a bit naive because I was thinking that on one leaf it would be mainly a monoclonal infection, and it turns out that it is not the case. Although because these observations are very fragmentary, one of the questions we had is what is this prevalence of these co-infections? Considering that we did not detect co-infections before, maybe there are other things we are missing in terms of the microbiome of the leaf. And an important point is what are the consequences on populations, because, well, as Professor Kumar said the first day, there are interactions within the microbiome, and so it's very difficult to predict what could be the influence and the effect of these co-infections on disease and on pathogen evolution. And so, based on this, altogether, this raises the issue of pathobiome. This is a new concept that is emerging, and we wanted to try to solve this puzzle, or at least try to contribute to advance it. And for this, we decided to take on a metagenomic approach, because we are dealing with strains that are taxonomically very close. We could not use a metabarcoding approach because the resolution is too low. And so we decided to go for a shotgun approach with long reads, and especially nanopore technology. This was for a number of reasons. One of the reasons is adaptive sampling. So I don't know if you are all familiar with nanopore sequencing, but the principle is that you have thousands of pores like this inserted in a membrane on a flow cell, and then you have the DNA molecule that goes through this pore, and the modification of the voltage between the two compartments delimited by the membrane, this voltage modification is then translated into a DNA sequence. The point is that in the sequencer, the sequencer is able to control the movement of the molecule inside the pore. So what you can do is insert in the sequencer a DNA sequence, and automatically, in real time, the first few hundred base pair of sequence are compared to the database, and based on the outcome, you can decide to either reject the molecule, okay, or you can keep sequencing. So this way, you are able to do enrichment or depletion in real time on the sequencer. So for us, it was interesting because we could try to deplete host genomes, so rice genome, and try to enrich for microbe genomes. So we did a lot of experiments to try to set up the system in the lab, using artificial inoculations like infiltration of rice leaves. I'm not going to go into the details because I don't have time, but the point is that we were very surprised. So we tested adaptive sampling, and overall adaptive sampling is very good at depleting rice read. So you see here there is no yellow, meaning that we could not detect any rice read. But if you look at here, adaptive sampling increases a little bit bacterial sequences, but not so much. So there are a number of reasons for this, and we can discuss it later on. In terms of read length, which is supposed to be a good strength of nanopore, it was okay, but we were a bit disappointed, and the main reason is probably because our extraction method for DNA is too harsh on the DNA, and it fragments it too much. In terms of coverage for the bacterial genome, you see we were very surprised because when you look at this graph here or here, we have a lot of DNA sequences from the bacteria, far more compared to the amount of sequences for the host. We already have quite a lot of reads from the bacteria, so this is very good. And we were very surprised about this. In terms of coverage of the genome, there is no major bias, so we thought it was very encouraging. And of course, one of the first questions we had after setting up the system is to look at what can we learn from this with field samples. So we use samples from Cambodia because we went sampling there in 2022, and so the usual cycle or the usual process we are doing when we go sampling in the field is to collect the samples, grind to try to have a lysate, and then spread on plate, and then through several cycles of isolation on plates, we try to save isolates in -80. And then we deploy molecular techniques in order to confirm the nature of the isolates. And we can also do pathotyping in order to understand the virulence profile of the strain. So this is the usual traditional way to do that, to characterize a pathogen. And we are wondering whether these metagenomic approaches could help us speed up the whole process. And so we started small with only 6 samples from 2 different locations in Cambodia, one in Battambang, one in Preavir. So 3 samples from Battambang, 3 samples from Preavir. Here you have the metadata about the samples, so we just noted the disease symptoms. And we also add the outcome of molecular characterization techniques. Here you can see the number of strains that were isolated from each sample. And so you see that 2 of them, we could isolate both XOC and XOO. So confirming that co-infection is indeed the case, at least in this situation. So for each sample, we took just a 4 centimeter leaf fragment, extracted DNA, and then proceeded for sequencing on the MinION. So at that time, we were still using MinION sequencing. So it's a small sequencer from Nanopore. And so here I don't have time to go into the detail, but this is one approach. One thing you can do with the data you obtain is to taxonomically assign the reads, meaning that essentially what you are doing is that for a million of reads, you are doing a blast against all available public genomes, microbial genomes on NCBI, and you try to identify the nature of the reads and assign them to a taxon. So I will not comment here, but there is very interesting information here. So another thing you can do with the reads is to assemble the genomes. So here you have an outlook of the output. So you see, for example, here the pink dots, they correspond to contigs, assembled reads, contigs, falling as assigned to the Xanthomonas oryzae species. So this is the sum of the length of the contigs assigned to the species. And what you see is that for most of the samples, we have about 5 megabase pair of sequences, which is more or less the size of a genome, meaning that we have near or complete genomes for each sample. And it's not only Xanthomonas oryzae, we have also Panthoea ananatis that is very high also, meaning that we have other bacterial genomes from these samples. So what can we do with these genomes? So for example, you can ask the question of where are these bacteria from? So in order to do that, what you can do is phylogenomics with metagenome assembled genomes. So you can you can construct a phylogenomic tree of all the public genomes in NCBI. So here there are about 400 genomes, and include also the metagenomes in order to position them within the diversity of the species. And so here, what we see is that the 3 metagenomes from one location, they cluster together with Chinese XOO strain, and it makes relatively good sense. And also the 2 other samples from the other location, they cluster separately, close to a strain we had sequenced before, which is from Vietnam, which also makes very sense because Vietnam is a neighboring country from Cambodia. And the last sample was XOC, and it clusters also with Asian strains. So to show you to illustrate the last application of the approach, I need to go back to what Nicolas talked about on the first day, TAL effectors. So TAL effectors, they are virulence proteins, okay? So they are translocated when the bacteria infect the plant. They are translocated inside the plant cell, and they act as bona fide transcription factor, meaning that they go into the nuclei, and they bind to very specific sequences on the host genome. I cannot go into details either, but what you need to keep in mind is that TAL effector sequences are very, the mechanism of DNA recognition by TAL effector is well enough understood so that we can, even based on the primary sequence of the TAL effector, we can predict with very good accuracy the DNA sequence that this transcription factor is going to recognize. And as Nicolas said on the first day, this TAL effector in Xanthomonas, and especially in Xanthomonas oryzae that is causing BLB on rice, these TAL effectors are very important, meaning that they target susceptibility genes, they target these genes and induce their expression. And if these genes are not induced, you have no disease. Okay, so in rice, there are major susceptibility genes that are sweet genes, so they encode for proteins that are involved in sugar transport. The point is that in each Xanthomonas oryzae, Pathova oryzae strain, you have at least one TAL effector that is going to target one of these genes. Okay, and if for some reason you manage to suppress induction of the gene by the TAL, you get resistance. Okay, and so one major approach recently to create resistance in rice has been to edit with CRISPR-Cas effector binding elements, meaning that you just change a sequence that is recognized by TAL effector, you prevent induction of the gene during infection, and then you get resistance. So other people have done that, we did that also. And so in general, strains, they have 1 to 2 effectors that target sweet genes. For example, they can have PTHXO2 that target sweet 13, and then have AVRXA7 that also target sweet 14. So in this case, if you want to have resistance, you need to edit both this EBE and this EBE, okay? But if you know the TAL effectors from a given strain, you can predict what sweet is going to target, and consequently you can act and deploy the appropriate varieties to have resistance. So we ask, okay, so in our metagenome assembled genomes from Xanthomonas oryzae, can we detect TAL effectors? And the answer is yes, although we add less of them than what we have in reference genomes, meaning that probably our genomes, they need to be perfected. They are not so good, but still. Okay, so I would finish. And so we not only we found TAL effectors, but we also could detect TAL effectors that are predicted to target both sweet 13 and sweet 14 genes. Okay, and so based on this, what we did is to use rice edited lines. Okay, that are edited in both sweet 13 and sweet 14 promoter EBEs, and we used a very standard assay for susceptibility of rice, which is leaf clipping. You just cut the tip of the leaf and you measure the length of the lesion. So this is a complex graph, but what you can see here is that, for example, a strain that relies on sweet 11 is virulent on all 3 genotypes of rice we tested: wild type, sweet 13 EBE edited only, and sweet 13, sweet 14 edited EBEs simultaneously. This strain is still virulent. But all the 4 strains from Cambodia, these strains were isolated from the samples we sequenced with nanopore. When we test them, the sweet 13, sweet 14 EBE edited lines, they are much less susceptible, meaning that they are resistant and that our prediction is correct. So I'm finished, and I'm not going to go into all this list because I guess we are running short of time. But what you can keep in mind is that with this approach, this is starting to give us ways to predict virulence genes, and based on this knowledge, to deploy resistant genes from the plants using a very easy method with a very small amount of material. And now what we are going to be doing is to move on to so we have already started using the Promethean. It's a larger machine that has a lot more throughput, and we're going to try to this time it was on Xanthomonas oryzae, but we're going to try to move on to other pathogens of rice to see whether we can replicate this approach. Thank you very much for your attention.
Thank you, Dr. Sebastian. Quick discussion, please.
Nice information you shared.
Thank you.
XOO and XOC, you mean to say they coexist on the same symptomatic plant?
Yeah.
Okay. Your nanopore sequencing you did on the symptomatic leaf, or?
Yeah, exactly. So we collected the leaf, we cut the leaf with symptoms, and then we extract DNA from that.
Okay.
Yeah.
In the symptomatic leaf, which fraction is dominating, XOO or XOC?
So that's an interesting question. So in this case, it was XOO. When you just look at the estimated number of copy of genomes. But in our experience, sometimes when you have samples with both XO and XOC based on molecular marker detection, so it's not quantitative. Sorry. So when you have this, very often when you try to isolate the strains, you get only XOC.
Okay.
So there could be some kind of an antagonisms between both pathovars within the tissues.
So why this is very important for us, because we normally go for screening in rice. We never bother about XOC. We bother about XOO.
Yeah, yeah. So
maybe we are ignoring XOC component in the whole syndrome on the leaf.
Yeah, some people that's something they are claiming. Some people they are claiming that BLS is unfairly considered as a minor disease. That's the first point, yes. And it could be that XOC somehow is helping XOO create BLB. So that's something to keep in mind. And I think also in terms of co-infections of strains from the same pathovar, that's something we were wondering is, for example, when you have strains that have, well, do you have interference between the strains? For example, you have some strains that are able to suppress resistant gene effect. So when you mix strains that are suppress resistance with a strain that is susceptible to this resistance, what happens? Because if one strain can suppress resistance, then this is something that needs to be taken into account when you screen resistant genes. Yeah.
Excellent. Very good information. Very crucial information.
Thank you.
Thank you, Doctor. Now, Dr. Deborata Laha from IIC is going to join in online. We'll wait for a few minutes. Now I invite Deborata Laha to present his topic on potential inositol phosphate messengers in augmenting plant immunity.
Thank you. Are you able to hear me? Can I share my slides?
Yes, yes, sir. Yes.
Okay. So I need permission to share my slides, so it is not there. No.
Now you try, sir.
Yeah, I'll try now again. Yes, left window screen. No, it's still not given. Can you please, I can rejoin the link.
Yeah, try just. Whole screen share. Whole screen. Please share the whole screen.
Oh, can you see my screen?
Yeah.
Okay.
Yeah.
Are you able to see my slides?
Just to open it. Open it, open it. Share it with us.
I did.
Now, you please share.
We see your file browser.
Okay, that's problematic actually. Let me try again. Select. Okay, now, yes, I think I'll be able to do this now. Are you able to see my slide?
Yeah, now you can switch to presentation mode. Yeah,
okay.
Perfect.
Okay, great. So I thank this organizing committee for this opportunity to, discuss some of this finding of we have. And I am extremely sorry that I could not manage to come in person because some sudden, immediate other situation that appeared. But nevertheless, let me start with, with the title of this, my talk today. This is potential of inositol phosphate, chemical messengers in augmenting plant immunity. So I think I do not need to introduce this audience about, different pathogens and, their role in their impact on plant growth, development, and yield. So this is, one of that herbivory, of course not the butterfly, but the larvae of this Piaris rapi, they can affect severely, the plant growth and development. And there are many pests and insects, they do that. The common way that plant encounters this pathogen infestation is through jasminic acid signaling pathway, and many of us, we know that jasminic acid, which is a plant defense hormone also involved in growth. This is perceived by the plant hormone receptor COI1. Under normal condition, the repressor protein JAS, it stays away from the COI1. However, when there is attack on plant by insects or necrotropic fungi, there is immediate increase in jasminic acid as well as the bioactive form jasminic isoleucine, which then acts as molecular glue and brings the repressor protein in contact with the COI1, which get ubiquitinated and degraded, and therefore, the jasmine is responsive dependent gene expression initiate, and plant becomes defensive against different pathogen. Now, what is interesting to note here that when the receptor, the COI1, along with the AX1, was co-purified and crystallized from insect cell, they identified in the core of the complex a high electron density, and later using NMR and mass spec, they have identified that this molecule is an inositol phosphate isomer. So let me quickly introduce about the inositol phosphate. So inositol is a cyclic polyol, and it can be phosphorylated at different positions, giving rise to soluble inositol phosphate such as IP3. I think we have, many of us heard about this molecule before. And the other thing is that the inositol can be incorporated into the membrane resident lipid, for instance, phosphatidylinositol, phosphatidylinositol 4,5-bisphosphate, and these are important signaling molecule in plants. So what we have studied in textbook that upon activation, the phospholipase C, when cleaves the head group of the PIP2, it produces soluble inositol phosphate IP3 messenger, which then diffuses through cytosol and binds to the receptor, which is a specific calcium channel localized in endoplasmic reticulum. Now, the binding of IP3 to its receptor leads to the conformational change, which result calcium efflux, and then it triggers further downstream signaling. Now, what is important to point here that such, this signaling pathway is only identified in metazoa, but this IP3 receptor could not be identified in any plant or in fungus genome. So therefore, how calcium signaling is regulated in plant is still a mystery. There are a lot of mechanism has been there, but IP3 mediated regulation of calcium signaling is not understood because they don't have the canonical receptor. Anyway, this IP3 can be further phosphorylated to IP4, IP5, and the fully phosphorylated inositol ring, which is known as inositol hexakis phosphate or IP6. The other name of this molecule is phytic acid. We might have heard about this name, phytic acid, because particularly those who are vegetarian, when they consume dal and legumes, beans, they are loaded with the IP6. They represent almost 2% of the dry weight. And in our gut, in acidic environment, they chelates iron, zinc, calcium, and leads to the mineral deficiency such as anemia. Therefore, one of the major efforts by plant breeder and geneticists is to generate plant or crops with low phytic acid. However, it has not been successful yet because, for instance, if we express heterologously XOPH, this is a type 3 effector from Xanthomonas that remove one phosphate from IP6 to IP5, and if you look at the consequence of the plant, so you can see on the left hand side the wild type plant expressing the GAP alone, and in the right hand two independent plant, these are XOPH expressing plant in translucent fusion with the GAP. There's severe effect in growth and development. Also, if you disturb the phytic acid metabolism by making knockout of IPQ1, you can see compared to the wild type, this is the consequence of the IPQ1 mutant plant. Not only that, if we suppress the expression or synthesis of phytic acid by suppressing the gene expression, you can see the potato plant become susceptible to the virus infection, suggesting that IP6 or phytic acid and its derivative are critical for controlling plant immunity, growth and development. So I mentioned that IP6 and its derivative. Yes, IP6 can have derivatives. These can be phosphorylated by a class of enzyme that is called ITPK, which make a pyrophosphate bond formation at position 5, which can then be further phosphorylated by VIase type kinase that makes IP7 to IP8, having additional diphosphate or pyrophosphate group at position 1. Now the difference between the inositol phosphate and pyrophosphate is that inositol pyrophosphate are also inositol phosphate species, but they are having one or more pyrophosphate species. Therefore, they are referred as inositol pyrophosphates, these things. And what we have learned that when you treat the plant, so now I come back to my original, the point of view that I have started with. If you treat the plant with jasminic acid, what you have realized, the inositol pyrophosphate such as IP8 level, it goes twofold higher, and that you see after 30 minutes of treatment, suggesting that this molecule might have a role in plant defense against the herbivory and necrotropic fungi. And then we talked with the IP8 and IP5 in the crystal in the structure of the COI1 AXO1 receptor. What we found that IP8 binds more efficiently to this COI1 receptor than the lower inositol phosphate such as IP5. And you can see that the highlighted in black, the histidine, arginine, and lysine, they can specifically interact with the IP8, but which is not possible for the IP5. Now, if we mutate this putative IP8 binding site of COI1, what you see, whereas the wild type COI1 and JAS1, upon treatment with the coronatine, which is the jasminic isolation analog, they interact nicely, which is very important for the further proteasomal degradation. However, if you mutate this IP8 binding residue, the consequence is, they do not interact anymore, suggesting that the binding of IP8 is important for recruiting to JAS repressor protein into the COI1 SCF complex. And furthermore, what we show that binding of inositol phosphate is important for this jasmine receptor complex formation, and we could show that it is not only the jasminic acid but also the VIase2 dependent IP8 production is very important. The coincidence detection of two structurally unrelated molecules, which is jasminic acid and inositol pyrophosphate IP8, is important for this COI-JAS receptor complex formation and degradation of the repressor, and for resulting the plant defense response. Now, the inositol phosphate role of inositol phosphate in plant immunity came in other perspective also. For instance, XOPH, which is a type 3 secretion, so Xanthomonas using type 3 secretion system, they release this effector protein, and as I mentioned before, that it hydrolyzes IP6 to IP5, and this is what happens in the plant, that IP6 is fully converted to IP5. You can see the red line, and I have shown you the phenotype, but if you look at the hypersensitive reaction, actually, the catalytic activity, the conversion of IP6 to IP5 is important for inducing this HR response. So we also know that when plant is attacked by pathogen, their growth is compromised, and we try to understand that is this same molecule is somehow, have a crosstalk between growth and development. The answer came up from a complete different perspective. I just quickly take you through before I finish my talk. So we all know that gibberellic acid is a growth promoting hormone, right? And what is interesting to know that in angiosperm, which is the flowering plant that evolved recently around 150 million years ago, it is the gibberellic acid is pursued by the receptor GID1, which polyubiquitinase, the master growth repressor DELA, and the subsequent degradation is important for inducing growth and development. Now, in bryophyte, what is interesting to know that they have also bioactive gibberellic acid. However, they don't have the canonical receptor, which is GID1. The question is how this DELA is regulated in this plant that doesn't have the canonical GID pathway. And the answer we got while we try to characterize a kinase that makes IP7 to IP8 in plant using Markenshire polymorpha as a model species. So what we have done, we have taken Markenshire polymorpha and we did CRISPR and we muted the kinase and we got independent knockout line, which is MPVH1 and 2. And you can see this is the wild plant which grows nicely, and the VIH mutant that are defective in IP8 production, that growth and development is severely affected. That can be rescued by the catalytic activity. That means the conversion of IP7 to IP8 is important for rescuing the growth. And we could show that it is not the cell area, but the slow growth phenotype is because of the cell division. I'm not showing all the data, but to get the mechanistic insight, when you try to identify the cellular target of this kinase, we identified DELA as a target, and we confirmed by pull down histohybrid BIPS experiment. And what you see that the mutant plant mimics the phenotype of the DELA overexpression line. So these are the two independent mutant line, and this is the overexpression line. So we hypothesize at the moment that in the wild type background, the kinase that makes IP7 to IP8 suppress DELA, therefore it induces growth. However, if you have a kinase mutant, then DELA is not able to be suppressed. It could not be suppressed, and therefore it suppress growth. If our hypothesis is true, then in this mutant background, if we suppress DELA, then it should, the growth should come back, right? And this is exactly what we see. So this is the wild type plant. This is an independent mutant line. And this is the same mutant line, but here the DELA level is suppressed, so the growth is rescued. And then what you also show, the DELA is accumulated in the IP8 deficient mutant line, whereas in the wild type level, you rarely detect these things. So the finding we got that the kinase makes IP7 to IP8. That IP8 binds to DELA. Experimentally, we have shown all of these details. I am not going to all this stuff. And then it gets polyubiquitinated and degraded, and it promotes growth and development. Interestingly, this mechanism that evolved around 450 million years ago looks like they are also present in the flowering plant. Right, so if you see, this is the Avidosis plant defective in IP8 production. This is VIH1, 2, PH and PHL mutant, and ITPK1 mutant. You can see that RGA, which is the homologue of Avidosis DELA, this level is very much accumulated in the plant defective in IP8 production. So this work we recently published, and we all know that DELA is encoded by the green revolution gene. So when did in 1960s? When they did the random mutagenesis, they found a lot of plants with improved yield. And one of the, after, in 1999 or 2000, when they performed the genome sequencing, it appeared that one of the maize product, which is reduced height, this is a gain of function of DELA. So we hypothesize that, our work can set light in a way that, so DELA overexpression line is already kind of short and dwarf. But then if you have an IP8 binding mutant that is not degradable upon gibberellic acid treatment, they can have more branch, more yield, and because DELA cannot be degraded by IP8, now in this plant, we can produce the IP level. Remember at the beginning I told you that how inositol pyrophosphate IP8 potentiate the interaction between COI1 and AX1 and therefore promotes the degradation of JAS. So I think this is one of the way we can look into the future, how this small molecule, the chemical messenger, can regulate, contribute to the plant immunity, as well as it also takes care of the growth, development, and yield. Yeah, and with this, I'd like to finish. This is my group who has done all this work and all the important funding agency who has supported the work. Thank you.
Thank you very much, Dr. Laha. Do other questions in the audience? Yes.
Quick question for you. You know,
Yes.
both phytic acid as well inositol phosphate, though it induces resistance in the plant system, or it helps in terms of jasminic acid and all, but it's an anti-nutritional factor.
Yeah, so that's an important point as I tried to highlight at the beginning. So the anti-nutrition point comes in the seed actually. So if you
Right.
have in a
Okay.
seed, but in the vegetative tissue, we need that phytic acid because otherwise if you make knockout of the kinase, or if you find a way to reduce the phytic acid level, the plant growth and development as well as immunity, everything is suppressed. So we need this molecule in plant. So if you want to have low phytic acid plant, I think the best way is that in a tissue-specific manner, suppression of the phytic acid, not overall these things. Yeah.
Because there is a major breeding program going on in TNAU as well in ICRISAT, looking at low phytase maize.
Yes, I'd like to discuss more in person. I hope I wish I could have been in person there.
Right.
Yes, so that is very important. Yes.
Another question would be, I come from nanotechnology background. We have heard of nano urea, nano DAP. Many times they say that there is a change in the phosphate transport system has altered when the nano DAP is sprayed. Do you have any comment on that?
So this is a good point actually. So I like to also mention. So recently there are work by from Michael Houghton laboratory in Zurich and in Geneva. My former PhD supervisor Gabriel Sharp in University of Bonn now, and also my laboratory. So what we found that this molecule IP6, IP7, IP8, they are sensor for phosphate status. So if you suppress the phosphate extracellular level, their level also goes down. And if you resupply with the phosphate, their level also goes back immediately actually. So they are highly sensitive. So at the end, if you think about inositol phosphate, this is nothing but a phosphate crowded inositol molecule, right? And looks like they have learned how to sense and trigger the phosphate starvation response pathways using this SPX PHR interaction.
Right.
So this is something also to be looked in more detail in future studies. And I think pathogen also use this pathway for establish the virulence. So I think this is a very important molecule to look at from different perspectives.
Thank you. I'll be in touch.
Okay, thank you.
Thank you.
1, 2. Is it working? No. Thank you very much, Dr. Laha. Maybe we can all join me in giving a round of applause for Dr. Laha before we go to break.
Thank you.
So now I heard it's a coffee break, a tea break. I don't know how long. 15 minutes? Okay. So see you in 15 minutes.
Good afternoon. And now I call upon Dr. B. Parameshwari, Principal Scientist, Hyderabad, Telangana, to present CRISPR-based diagnosis tools for detection of plant virus in quarantine. Please. Madam, please take only 15 minutes, including discussion.
Respected Chairperson and distinguished scientists from India and friends, good morning to one and all. Slide. Plant viruses are causing important diseases in agriculture and horticultural crops by reducing the yield and product. Here in case of plant viral diseases, diagnostics is very much important. Here NBPJ regional station Hyderabad is the quarantine station. In case of NBPGR is involved in plant germplasm exchange. Here divisions are different from normal these things like division of exploration, division of evaluation, division of conservation and division of quarantine. Our station NBPGR regional station Hyderabad is coming under division of plant quarantine. It is established in 1985 particularly for the ICRISAT, quarantine activities of the ICRISAT, import as well as the export. Then after in 1997, that service extended to other public, private and international institutions located in southern India. Here in case of plant virus detection, we are following the different technique, namely normal invected assay based on symptoms, then electron microscopic observation, serological based detection ELISA, then nucleic acid based detection. Here in quarantine, actually Hyderabad is the seed cup. Many seed companies are located in Hyderabad as well as Bangalore. We are the different plant protection scientists, plant pathology, entomology and nematologists are working there. So we have to do the rapid crude set based detection in field itself. So I have carried out the RPA based detection along with the CRISPR. So in crude sap itself, without isolation of DNA or RNA, we can identify in the on-site detection. In field itself, from ICRISAT also we used to get the samples for export as well as import of material from other ICRISAT gene banks, and as well as for Svalbard also. Svalbard is located in Norway. For the germplasm exchange also we are involving. These are the different isothermal nucleic acid amplification methods, that is LAMP, RPA, etc. Here no need of PCR in case of isothermal amplification is method. Here only the temperature is 37 to 42 degree. We have to standardize in between this temperature with different incubation time. Here for sensitivity is high compared to the PCR. We can per reaction 1 to 10 copies we can diagnose. So in case of RPA, this is isothermal. PCR different cycling condition and temperature is different. Here same temperature means 37 degree we can do with dry heat bath. In case of RPA, then crude sap based detection is possible in this method. For RPA, we are using TwistTX companies this RPA kit. This is developed by TwistTX UK company. Here in India, dealer is the BioLink company. This developed by the Pippenberg kit in 2006. Here mainly recombinase and polymerase enzymes are there to detect any plant pathogen in isothermal conditions. Using only dry heat bath, here no need of PCR. This is the amplification method. Actually in case of our quarantine station, we have to identify different viruses, pathogen, fungi. What are the different methods? Here I am working, my specialization is plant virology. In groundnut, peanut mottle virus, peanut stripe virus and stunt virus are there. These are quarantine pests. Then some viruses are seed transmitted also. So these are very important. So on-site detection, we have developed RPA method. In case of RPA, the primary length is 30 to 35 nucleotide. To increase the sensitivity, the primer length is more, the amplicon size is less. So 100 to normally 300 base pair, it can go up to 500 base pair also. Then here CG content should be 30 to 70%. Then we have to validate the RPA primers. In case of RPA reaction setup, they are provided with the rehydration buffer. Then forward and reverse primers we have to develop. For example, core protein gene is there. In that we have to develop RPA primers. That should be 30 to 35 nucleotide length long. Then that one pellet will be there. That enzyme we have to dissolve it, and magnesium ester we have to add 1.25 microliters to stop the reaction. Here only temperature is 37 to 42 degree. We have to standardize in which temperature that RPA primer is able to amplify. Then incubation time also we have to standardize here. In case of RNA viruses, we have to, in crude SAP I am telling, after grinding, in case of RNA viruses, we have to add the reverse transcriptase enzyme as well as the RNase inhibitor for the RNA viruses. Like peanut mottle virus is RNA virus. Some other virus, DNA virus is there, means no need of adding the reverse transcriptase or RNase inhibitor. Here incubation time is 37 degree. For 20 minutes we got amplification for peanut mottle virus. That is, in core protein region is around 900 base pair. In that RPA primer is 270 to 300 base pair length. Here you can see these infected plants we got from the quarantine material grown at the ICRISAT separate post-entry quarantine facility is there. So we are NBBGR scientists, we are having one room for every month, weekly once like that we have to go and check all the materials. Any fungi, bacteria, virus, so that we have isolated and maintained at the French bean. Then we have amplified through the core protein specific primer of peanut mottle virus. We got the amplification. Then using 2 RPA primers we designed. Both RPA primers we got the result. You can see 300 base pair and less than that one. So that second RPA primers we have used for further purpose and sequence and submit to GenBank also. Then cross reactivity also checked. So further for CRISPR reaction, this one we have used here. First we have to standardize with RNA, cDNA. Then crude SAP last we have to go. In crude SAP different buffers we have to use for grinding. Here you can see in crude SAP these are the 10 different buffers we have used in different temperature and incubation time. In case of crude SAP we have used one normal sodium NOH and NOH EDTA 1:1%. We got good banding, one normal NOH. So further we have used that crude SAP of this one for grinding, and we have on-sitely we have detected that peanut mottle virus. You can see here. In case of crude SAP, in case of PCR, you are not getting any result. But in case of RT-RPA with crude SAP, you are getting the 300 base pair band of peanut mottle virus. So we have done comparison of RT-PCR and RT-RPA. Here for PCR, we have given less only 2 lakhs, but for gradient PCR, it will go around 4 to 5 lakhs also. Dry heat bath, the cost is around 20,000, we kept 42,000. So based on this everything, for RNA virus means we have to use RNA isolation kit or a conventional method. Here in case of with crude SAP, that 290 rupees per reaction in case of RPA. In case of DNA virus, it is around 250 rupees only. Then we have validated that RPA technique in the field collected samples from different places. From our lab, this is the first report of RPA based detection of peanut mottle virus. Then similarly for tobacco streak virus also, I got some SERB Power Fellowship project and Women Excellence project from SERB. So in that we have done tobacco streak virus in sunflower also. Then further seed transmission of tobacco streak virus also we have done using the same RT-PCR followed by RT-RPA, then real-time PCR also. Then herely for sugarcane, before joining NBPGR, I was working in sugarcane Institute, Karnal also regional station. So that one project transferred to NBPGR. So in that RPA based detection of sugarcane bacilliform virus also, because sugarcane also vegetatively propagated, the sugarcane bacilliform virus also, it is important pathogen. Early it was quarantine pathogen, now it is present in India also. Then similarly in case of chili, cucumber mosaic virus, one of my MSc student, we are having the medium term module at Hyderabad also. Many germplasm we are having. So we have screened around 50 germplasm. Along with we have developed RPA also for this cucumber mosaic virus. Similarly for peanut stripe stunt, these are quarantine pathogen. For that also we are doing. So these are the technologies accepted by the ICR and certificate provided by the ICR in recent Foundation Day of ICR in this year. Similarly, after RPA, we are combining RPA with the CRISPR. In case of CRISPR, different Cas-associated proteins we are using. Here, what is the important means sensitivity is increasing. Here also crude sap, but for standardization we have to do RNA, DNA, everything. Finally, in crude sap only we are identifying. Here we are using Cas12a in case of peanut mottle virus. This will cleave the single stranded DNA reporter. Here you can see this is the peanut mottle infected plant. We are doing RNA extraction. This is for standardization. Then we are converting to complementary DNA. In case of DNA virus, directly we can do it. Then RPA we have to standardize for 37 degree for 15 minutes. After that, we have to add the guide RNA is there. That subsequently I will tell you how to design guide RNA. Then Cas12a enzyme is there. After adding this Cas12a guide RNA, then one single stranded reporter is there. We are detecting the presence of virus through fluorescent technique or through lateral flow assay. In case of Cas12a, that PAM sequence is TTTV. So V can be ACRG. So after that PAM sequence, we are taking 24 nucleotide length. After that spacer sequence is required. That is guide RNA. RNA sequence that is conserved is there. I will show you here. This is the scaffold sequence. This we have to add before the spacer sequence. Spacer sequence will come within the code protein gene only, 24 nucleotide. Before this we have to add this. Then only it will form the loop-like structure. Then it will bind guide RNA. Here you can see that RPA primer I designed here. This one, this is forward reverse. In between you have to design the guide RNA between this RPA primer sequence. This TTTG. This is the PAM sequence. After that we have taken 24 nucleotide, but it should be RNA form. We have to convert to RNA form. We have to add the scaffold conserved sequences to increase the binding of the guide RNA with the target sequence to form the CRISPR-Cas12a complex. Here you can see CRISPR-Cas12a complex. This is the PAM sequence. These will go and bind here. Then we will add one single stranded reporter, that is TTT. In case of lateral flow, that is FAM and biotin linked. Then this complex will formed, then it will go on staggerly, collateral cleavage will happen. It will cleave the single stranded reporter. Fluorescence means different reporter is there, lateral flow different reporter is there. This is the RPA region. Here you can see the PAM sequence, TTTG. This sequence we have taken as guide RNA. But it should be in RNA form. Then here scaffold sequence you have to add. So these are the different guide RNAs designed for the peanut mottle virus. So this is the single stranded probe for the fluorescent reporter. FAM TTATT PHQ. FAM and biotin lateral flow reporter for lateral flow assay. In case of RPA-coupled CRISPR-Cas diagnostics, first we have to carry out the RPA reaction. By adding the forward reverse buffer water, the total concentration should be, one pellet will be there that we can use for 4 to 5 reaction. After that CRISPR complex and lateral flow stick also we can, by fluorescence we have identified the peanut mottle virus. By visual only, we have to standardize single stranded report, guide RNA, everything. Similarly for lateral flow also, here you can see this is the control band. This is test band. So you can see the result for the on-site detection of the peanut mottle virus. Similarly for the other cucumber mosaic virus of tomato and chili also we have done. In future multiplex, in case of groundnut more than one virus are quarantine importance in India, peanut stripe, mottle and stunt also. So now we are doing multiplexing of through RPA coupled CRISPR sequence. Thank you to the organizers for giving this opportunity.
Thank you very much, Dr. Paramisawari. I'm wondering whether there are any questions in the audience. Sure.
Yeah, it's really you integrated many technologies, including CRISPR-Cas technology. All along, I have been really wondering why we need to really detect the virus in the symptomatic plant to an annual crop. I always have the view, you may differ from my view, we need to intercept the disease on the seed or on the planting material which is not showing any symptoms.
Seed also we have standardized for tobacco tree.
See, many times people use the symptomatic tissues for this kind of technology-driven interpretations, which I feel it's a futile exercise because viral disease at large, the management is by and large same kind of thing if you are going for a pre-harvest management in the field, either vector control or one question will come that might hurt us also. So what?
First we have to do it in leaf only, sir. For seed also we have
The standardization as a positive control is an integral part of any project.
But
When
PEQ
it comes to open field, in a societal application, probably we need to graduate beyond what have been doing with the symptomatic plant. That's my
actually in
comment.
seed that virus titer is very less also. In case of our quarantine, after releasing seeds, post-entry quarantine another is there. So we will go and inspect there. Na? All the time in seed it is not showing, titer is very less.
Right. So actually the thing is, people are really struggling to find a commercial partner to take this technology to the next level. It's stuck at TRL 5 or at the max TRL 6. It will never go beyond that. This is a problem. Even Agidaya company who is promoting, you just go and see people buy only for academic interest, not for any field application to solve the farmer's problem. What I'm trying to say, you are in a very good position in a NPPGR. Try to develop a technology for a seed which arrived our country, because I know we discussed also. So these technologies have a great value in those materials. Yeah, that's my opinion. Look, it's not to
for
find
tobacco
fault with
stand-
somebody, but this is my observation.
Streak virus we standardize
Just a
in
quick
streak
question
virus.
because time is running out. What is the resilient time of the diagnostic tool? Whatever you say, will it stay good for 1 month, 1 year?
No, it will say sir, sensitivity is high, sir, RPA.
Sensitivity is fine, but how long do you have to protect under refrigerated condition?
Minus 20 we have to keep, sir.
Okay.
When we are taking to field, that we have to take with that minus 20 box.
Okay. Another question would be, there will be variants, no? You're looking at one particular strain, but there may be other variants in the, how do you resolve this?
So now sequencing also we are doing, sir.
Okay.
If within
But
that,
it takes time, no?
It is take time, sir. But tobacco streak virus we have done it from seed itself. That is my PhD student work.
Okay.
Already through RPA we have done
But
it,
it's
sir.
well done. Excellent.
So lateral flow.
Yeah, I have some answer for you in my presentation.
Thank you very much. Thank you for your talk. Congratulations. So now it's our pleasure to welcome Dr. Subramanian. So I think it's an exciting talk about nanoparticles and their application in agriculture.
Yeah, sure. Okay, good afternoon to all of you. First of all, I have to thank TNA administration and the friends from France for organizing such a beautiful, international type of workshop. I really enjoyed for the past several hours, and it is very exciting. It's a small group, but very smart group, and I learned a lot for me to take it forward. I just flew from Delhi. Yesterday we had a discussion about exactly what we discuss here. Some of the diagnostic tools, immediate attention is required. So my presentation will focus on some of the emerging technologies for pathologists, plant protection scientists. Where do we go? And I am also a policy maker for the country. I am a spokesperson for nanotechnology in agriculture. Many of the decisions what the government of India is taking, it is from me. So certainly my, whatever I am saying, you have to carefully listen. So possibly in the evening or afternoon roundtable conference, it will be helpful for taking it next level. Can I have my slides? So it is certainly a good one, thanks to Dr. Sendil Raja. I don't know whether you know the meaning of Raja. Raja is a king. He's the king of the event. And another king would be our Frederick, another king from France. Both dialogued very well, very well organized, and each and every presentation, though from youngsters, I love to listen a lot. Though I come from soil science background, I learn a lot from the other areas. I think you have to change the background, huh? Like why it is happening like this. Okay, Apple. Oh, no. In that case, I'll pass on PDF so that you cannot see this slide. Maybe the first slide. Next slide will be all right. Next slide will be all right. Yes, please. No, all the slides will be like this. Maybe I share PDF format. Sorry about that. I'll keep on speaking. PDF. Right. Okay, I shared the PDF. So 3 points I just want to flag off before get into my presentation. The first would be I am going to show some of the update on innovative technologies which will be useful for both. In fact, all the participants in the hall will be benefited from my talk. That is number one. And number two would be the country like India. We have got so many emerging diseases, and the country is looking at nano is one of the solution. Nano pesticides emerging, so we have a regulatory framework in place, and the people looking at nano pesticides, next generation technologies. So that is number two. Number three, certainly in the roundtable conference or whatever we are going to discuss post lunch, so we'll take forward next level. So TNAU has a beautiful nanotechnology center as a founder head in 2010. In 15 years, we established a good center, and that center is a referral center for the country itself. And that is a center responsible for development of nano fertilizers in the country. Currently, we produce 85 million liters of nano fertilizers, which cover 40 million acres in the country, which is going to save about 1.48 million tons of urea, as well 0.48 million tons of diammonium phosphate. So it's all from TNAU only. TNAU as a researcher, we started working on that. Now we have come to this level. Certainly, we can help you in developing nanopesticide, which is most effective, less active ingredient, more effective. That type of technology I am going to show. So my presentation will cover, I'll say what is nanotech, because most of you would not know what is nano. Nano is very small, 1 billionth of a meter. 1 billionth of a meter, it is equivalent to, I always, go to the next slide please. So I was a director of research of Tamil Nadu Agriculture University. I always encourage scientists to look at emerging technology rather than the traditional way of doing. So this is the first slide. Second, how do I change? Okay, I'll change it myself. Sorry, it's not. Doctor, it's not changing. Can you play for me? You have to change it. Yeah, okay. 1 billionth of a meter, that is in terms of crore, 100 crore. Always I refer Indian map. India has 146 crore, that is 1,460 million people in India, most populous country in the world. So you imagine that entire population of India is squeezed in 1 meter length, each Indian is the size of a nanoparticle. So why nano is very important? Nano is very important because of the surface area. So you imagine that one cube undergoes a division, 24 times a division, you achieve nano-sized particle. Because of the nano size is so small, surface area is huge, you will be able to detect the disease much before symptom appears. That's what he asked. Well before symptom appears, you will be able to detect the disease. That is the advantage. Typically, every one of you carry a simple device called mobile, which has a nano device in it. So that nano device is referred as a silicon chip, which is very small, but it carries many information. All of you are aware that mobile is part and parcel of our system itself. You will forget many of your vital organs, not the mobile. So mobile is so powerful, so do nanotechnology in agriculture. So I closely work with Dr. M.S. Swaminathan. He is a father of green revolution in the country. He is responsible for the increase in food grain production by 7-fold. So I worked with him. I presented in 2015. He provoked me to think that nano would be the evergreen revolution. So I was very impressed by his conversation. That's why I goes on. There are 4 areas in which pathologists and entomologists can work in this area. One of the important, I was very impressed with the previous speaker presentation. It is very, very important that pathologists and entomologists should come up with the diagnostic tools. It is not gone are those days, you traditionally do all the tests and it takes several weeks to identify the problem. That should not be the case. You need an on-spot decision how it can be achieved. There are 6 diagnostic tools I am going to suggest. One is, of course, we'll go one after the other. Some of the nanomaterials can be used as a fungicide, pesticide. Some of the nanomaterial which I am referring, it is derived from the plant itself. Plant-derived nanomaterial can be used as a pesticide for plant itself. So that type of nanomaterial we do. And a smart delivery of input can be delivered like a medicine. Exactly where it is required, we can deliver. So nanodiagnosis is number 1. Earlier speaker also presented this. Only interventions of nanotechnologies here is the nanoparticle. We use gold nanoparticle or silver nanoparticle conjugated with the DNA, conjugated with antibodies, so that each and every reaction analyte is resolved. So typically for viral disease, one of our colleagues in NRC Banana, Dr. Selvarajan, he's developed a dipstick method where you simply take the leaf extract, put the dipstick, 1 minute you do you know what it is. So that type of quick analysis is possible. Here, as I mentioned, the surface area matters a lot. That is why we will be able to resolve well before symptom appears. So the diagnostic kits are aimed at 3 ways. So we need to identify the analyte, we have to transduce the analyte, and we have to develop as a sensor. So the sensor development has 5 stages. We have to identify the analyte, we have to absorb biosorption should be done in a substrate, then transduction to be done. It becomes an electrical signal so that common men understand whether you have a disease or not. Typically, you are COVID-19 identified with this type of device only. So that type of device is required. The second important area is a biochip, where hundreds of samples can be analyzed together. I have seen one such device in Taiwan in 2012, where hundreds of samples can be analyzed at once. So when you put the biochip into the machine, you get different spots. The green spot, no disease; yellow spot, mild disease; and red spot, immediate action is required. That type of strategies we need to evolve. Recently, we wrote one review with Sendil Raja. So that review focuses on quantum dots. Quantum dots are a cluster of electrons. We can synthesize quantum dot. We have a capability. These quantum dots can be put it in the sample, and a laser beam hit the sample, you get the excitation. The color change will lead to immediate direction of disease. We have written a quantum dot review, which is given below, so you can refer to that paper for more information. Somebody was mentioning about a organism called amylolichifacins. It releases certain compound that kills other pathogens. So eNose is very important technology. eNose is electronic nose technology, where any infection that are happening in the seed or any plant, the volatiles are produced. That's called VOC. Volatiles are hundreds and thousands of volatiles are produced. We have identified certain volatile which is specific to seed infection. That's called acetaldehyde. Sometimes it is ethanol hexanol, those are the compound that comes out. These are the reference molecule or spectral signatures. So by doing this, we'll be able to analyze. So what we do, we capture the VOC, we put it in the ENOS device, and you get the result. Within 1 minute, you get this. So we convert the biological signal into electrical signal. That's called transduction we do. We can do that quickly. That's another technology is closely with the e-tank technology. E-tank means you are not consuming the seed. What we do, we put the seed, infected seed in the water, and the leachate is taken. A drop of leachate is put it in the nanomatrix, where we can get this electrical signal. You compare the traditional method versus our method. Traditional method, what seed technologists usually do, they look at the electrical conductivity as a measure of infection. So the electrical conductivity, if you measure in a period of 6 months, it is increased by 0.6 to 0.8. If you look at the nano device, which is very accurate, it goes 3.6 to 1.68, which means each and every infection which are happening in the seed, which can be resolved. It's a e-tank technology. So very good technology. These are the things TNA has done. Lots of work on that. Another important pest in the field is the fall armyworm. Fall armyworm, when I was the director of research, the entire entomology team worked only on fall armyworm. That's a major, major pest. So we didn't know what to do with this. So we started working on AI technology. We also worked on some of the imaging technology. One of the powerful imaging, so after our entomologists say the fall armyworm is very difficult to control, I asked them to bring the egg mass. When we put it in the scanning electron microscope, we see that, so these are the egg mass, which is toughly covered with, lots of filaments, lots of fibrous material. That's why this particular pest could not be controlled. So there are 3-4 imaging can be done. Field imaging, thermal imaging, hyperspectral imaging, multispectral imaging. You do all the imaging together, put it in the AI tool, convert into a signal that your field is infected. So same thing can be used for disease as well. So this is another technology which is emerging, very good technology that be done. Another important most of the technologies, of course, it's initiated in TNAU or evolved in TNAU as well. One of the another technology is a paste and peel technology. This is very, very important for Indian agriculture. In fact, the world is looking at the pesticide residues a major problem. Suppose you get an apple, whether apple is safe to eat, whether there is a residue present. So TNA has come up with the paste and peel technology. What we do, we take a small paper strip. It's nothing, fugitive. It is a small paper strip which is loaded with nanomaterial. This nanomaterial is covered with a specific compound which can attract a specific pesticide. So what we do, we touch the strip, paper strip on the surface of the fruit. Within few seconds, the surface residue get transferred into this. Then we take this strip to the another machine called surface enhanced Raman spectroscopy. In 1 minute, diagnosis is done. Very good work has been done. Lots of papers have been published in this area. So far we identified these are the important pesticide, fungicides have been resolved with the paste and peel technology. What is the complexity in the system is that if you look at local market, you get a number of pesticides which is banned long back. So certainly this device has to be advanced by loading several nanomaterials together so that many diseases, many residues can be identified. The surface residues are more important than the internal residue. So TNAU has worked with ICR very closely. We found that if the surface residues are resolved, your fruits or vegetables are safer. So several technologies have been done. That's why we targeted that. There is a nano lure has been developed by both the TNAU as well as ICR. There is a ICR institute called the National Bureau of Agriculture Important Insects in Bengaluru. They developed a nano glue. A glue to attract fruit fly in the field. So compared to regular, lure, which lasts in a few minutes time because they use eugenol, which evaporates quickly, the nano glue stays for a long time, attract female and male insects as well. So yesterday also we had a discussion, and TNAU has come with a lure for rice stem borer. There is Sirboboga institutus, they have done it. So these are the diagnostic tools are required for both entomologists, pathologists, nematologists, all plant protection scientists. This is very important. Even nematode we can identify. These are the things. Secondly, I just will highlight 2 or 3 nanomaterial which can be used. Don't raise the flag. I have to complete. I will complete it. Okay, nanoparticles for plant disease management, even plant, pest, insect pest as well. There are 3 nanomaterials. Why nanopesticides are more effective than conventional pesticide? This is the illustration indicates when the droplet size is so small, absorption is faster. We recently found that when the spray of nano urea versus regular urea takes 2 hours. Regular urea or conventional urea takes about 24 hours to get absorbed. So do DAP. So likewise, when pesticides are developed in nano form, the absorption would be quick and the effectiveness would be very high. Yesterday they presented in Delhi indicating that the pesticide utilized by the plant for the protection, it is 1%. 99% goes to the environment. So that can be minimized by this. One of the very interesting nanoparticle which has a plant protection effect is the zinc oxide. Zinc oxide is very easy to, many of the cosmetics has zinc oxide in it. You simply coat 1 gram per kg of seed is good enough to control most of the seed-borne pathogens like macrophomina. Our friend asked about rhizoctonia, that can be controlled with this particular thing. So we have shown that there is a lipid peroxidation goes down when it is treated with zinc oxide. Another interesting nanomaterial is a silica. Silica can be derived from plant. That's why I said nanomaterials can be derived from plant and used as a pesticide for the same plant. For example, rice and sugarcane, these are the silica-rich plants. So we have the methodology to develop nanosilica and it can be used. Recently I wrote a review on this. This review carries n number of protection activity, again diseases, pests and nematodes, abiotic stress, biotic stress, n number of stress tolerance could be achieved with silica. As you all know, silica is also induced resistance, SAR. Systemic acquired resistance is possible with the help of this. Silica could be a very good vehicle. Silica can be a vehicle to deliver specific biomolecule. We already demonstrated 2 biomolecules. One is azadirachtin, another one is anoscomosin. Both molecules can be delivered. We wrote a paper also. So that, that helps in terms of inducing the resistance, also minimizing the transmission of virus. So as powdery mildew was spoken a lot, we can control with the silver nanoparticle. It's easily synthesized. It can be sprayed and control the powdery mildew. A storage pest, still when we were student, Celfos tablet aluminum phosphide was used as a protectant. Still it is used as a protectant. So no alternative. With nanotech, there are several eco-friendly nanomaterials have been identified for protection of storage pest. Nanoherbicide can be done. Some of the metabolites, plant-derived metabolites, as our friend says about some of the plants have the deterrents. So those materials can be extracted and developed as a nano formulation. That's why we got a patent for nanoherbicide that can be done. Smart delivery system, there are 2 smart delivery system, I would say. One of the easiest and delivery system is the nanofiber technology, which involves you can develop nanofibers with electro spinning machine. TNAU has 2 machines. Currently I work with the industry. That industry also has the machine. This is a very fantastic technology where polymer is converted to fiber. You can load any pesticide of interest. You can imidacloprid, tuberculosol, any pesticide, anything can be done. You can use this technology. One of the power of this technology is that the nanofiber has a tremendous surface area. 1 gram of nanofiber can connect Delhi with Coimbatore. 1 gram. That is the beauty of this technology. So we can use it. So we already demonstrated tuberculosol is one of the fungicide can be loaded and delivered through this. And the BT, which is commonly used as a plant protection agent, particularly for bollworms. And recently there is a problem of high temperature, climate change, The BT which we evolved earlier is not working anymore. We need encapsulation, we need nanotechnology to protect or improve the resilient time. We can also mimic some of the things. We learn from the nature and we do that. The honeybees are very important for pollination. There is a bee board is there in the country and ICR as well, Government of India promoting honeybee as an agent to improve the productivity of crops. But rearing honeybee is a rare, difficult task because of the, sunflower is very frequently predated by birds. This is a major problem. So we developed a nano lure for attracting honeybees. Without flower, you attract honeybees. So that's lure we developed already with the help of this. So final point for this forum, I had an opportunity to present the regulatory guidelines, the book which I will share to Sendil Raja, all of you can have it. This is it's a public domain. This is a book that is used for regulatory framework for any of the nano products that are developed in the country. I presented in the Parliamentary Standing Committee. So that particular book has information about all the OECD guidelines. These are the numbers referring to OECD guidelines. There are about 25 to 30 tests have to be done to make sure that the nano is safer to the environment. So these are my all my publications, all my patents, and you can Google. People call me KSS. If you do that, you get lots of information. Thank you very much for the opportunity. I exceeded my time little bit for the benefit of all the participants in the crowd. Thank you very much. You take my number. If you want to communicate with me, you can do it. Any question at this point, I can also address. Thank you.
Very informative talk, sir.
Okay.
Thank you, sir. The only concern for as a researcher is we are really afraid we will face the consequences of this, what is called as regulatory regimes, because I don't think in the near future regulators are going to approve any nano things for crop protection.
Okay.
What is your comment on that?
Yeah, okay. Yes, we are alarmed. I remember I presented nano views in 2011. Everybody was racing against me. In 2017 we did that. None other than the Prime Minister of India said nano fertilizer can go first. Now we have reached this level. As I mentioned, 85 million liters of nano fertilizers have come. So nano fertilizers compared to pesticide, fertilizers are more used in terms of volume. What you asked, the same thing asked me yesterday also. One of the critical points we want to address in nano pesticide is the active ingredient. The active ingredient, what is used for a particular pest to be controlled for a ETL, economic threshold level, it is only 1% of the total volume of pesticide. That is what people say it. So in that case, 99% goes to waste. So why do we do that? So we try to do same pesticide, which is already tested beyond doubt, this pesticide molecule, imidacloprid or any molecule of interest, that can be loaded into nanocarrier. Already there are soft nanocarriers which I also mentioned. You do that and deliver it. So nanocarrier is safe. The pesticide which identifies is also safe. When you conjugate together, the delivery vehicle, certainly it will reach the target. One of the alarming point which people say that because the size is so small, it may affect the environment, may affect unintentionally the user and all. So we evolve strategy for this.
Yeah. Even the toxicology data, I don't think we can go with the same what is there for other molecules.
Okay, oh, right.
We need to draft another scheme of things for...
Yeah, that's what we made a suggestion yesterday.
That's what, since you are with the government, you can really support government, the SOPs for nano formulation.
For pesticide, yes. We'll do that.
You please do it, sir, so that
number
of papers are
actually, yesterday we had a discussion with the CABRC as well. So certainly we'll take forward your point. Thank you very much. Any other point? It's Dr. Govindraj from ICRISAT.
Thank you, sir. Thank you for your presentation. I'm audible?
Yes.
Okay. we get rare opportunity to ask the question our professors, when I was...
No.
I just commenting, it's just a great opportunity to learn again in the new technology. But we are working in the food system, the transforming the food system. Whatever we discover the technology that boosts the food production, but when it comes to the application implementation country like India,
Okay.
Where the marginalized farmers population.
Okay.
So what kind of thinking as the government, as you are one of the advisors, how this can be routed through any subsidized program? That's the scalability question. And the second one is, the upstream question is, how this nanoparticle is going to degenerate within the plant system.
Right.
And what are the nano carrier is recommended to reduce the molecular interaction within the plant system.
I think to answer all your question, I have to give another hour. That is not possible. To make it very simple, what I would say that your questions are very relevant. What I would say is the scalability is a typical example, is a fertilizer.
Sir, excuse me. Sir, actually, we are waiting for the annual function. Vice Chancellor is going to come now.
Yeah, Vice Chancellor is our friend only. Vice Chancellor, he will be very happy. Okay, because acting, please come, sir. Please come, sir. I have to say in front of you only. Okay, please come. I am very, no, sir, Dr. Wagner. So, because acting Vice Chancellor is there, to conclude, please sit here, sir. So, I conclude in front of the acting Vice Chancellor, there are three key messages